Good morning, everyone. Please, some of you it's here online can log to see. João, can you hear me? I hope it's working. Okay, but welcome for the last day of the winter school. Unfortunately, Wellington, it's not going to be with us today. He abandoned the ship already. Have some other commitments. And today the work will be much more related to the antimicrobial peptides, some of bioactive peptides produced by the lactic acid bacteria when we ferment some food products, their application. So we'll be more or less around this. So the first presentation for today going to be on the Bacteriocins again. Obviously, we need to repeat some of the concepts because not everyone is every day with us. But, well, let's start. So we already talked about the variety of the antimicrobial compounds produced by the lactic acid bacteria. But one of them was really paying our attention is antimicrobial peptides, as called the Bacteriocins, because these powerful metabolites produced by not only by lactic acid bacteria, but with all type of bacteria, in fact, was very interesting because they can actually modulate the quality, not only the microbiological quality, but the organoleptic properties, because they can actually inhibit some of the other microorganisms in the food system. And in this way, they are responsible not only for the safety of the food products, but they can modulate even the other properties of the products because of the fact that they are very active against all the organoleptic properties because they can modulate the presence of other microorganisms. But the interesting point is that we're actually looking for the option that we can use the microorganisms with more than one properties because, well, we have the starter cultures. They need to conduct the fermentation process. Some of these are associated with their technological properties. As well, some of these microorganisms involved in the food fermentation process, they can have their beneficial properties. We heard a lot of presentations related to the probiotics properties of microorganisms, but probiotic properties are different from the technological properties, and they are giving additional benefits to the fermented food products because of presence of these live microorganisms in fermented food products. Then beneficial and probiotic properties at the same time, they're different things. They're not the equal because probiotic needs to give the benefits to the host, and this need to be live organism that giving the benefits to the host. But at the same time, beneficial properties could be different metabolites produced by this microorganism because this is a completely two different things. In bacteriocin production as well, it's a different point because the production of these antimicrobial metabolites by the microorganisms present in the food system are involved in the safety of the food products, and as I mentioned, they can be modulating antimicrobial population in the product, and this way they can affect actually technological properties. So everything actually is very linked and related to one to another one. So, and forming a concept 4-in-1. I mean, most of you, especially the women, know what's mean shampoo and conditioner 2-in-1, 3-in-1. But now we're having 4-in-1. We want a microorganism that can do all these things. This will be possible. Is this just a dream or it's a reality? So we want to have a microorganism that could be a starter culture, microorganism that will be having a probiotic property, so we can apply it. And via the food products, we can deliver this live organism to the consumer that will perform beneficial health stimulating properties in the host when go to the host. We want a microorganism that can have the beneficial properties production of, let's say, of GABA. That will be good to have it for the consumer, produced by the same microorganism, and this same microorganism to produce the bacteriocins that will actually inhibit the pathogens. Well, it's not an easy task, but I think will be possible with the right microorganism. But again, nothing wrong to have a dream and to think what we can actually apply. By definition, bacteriocins, again, they are produced by ribosomal system. They are proteins, small proteins. Very often, even we need to call them polypeptide because they're not even classified as a protein, just a polypeptide, too small. And they are active against closely related microorganisms. So we saw the several presentations climbing activity of bacteriocins against produced by the lactic acid bacteria that are gram-positive, with activity against gram-negative. Well, this is a good idea. We have some evidence to believe that this can happen. This could be a fact. However, we need to be very careful when we do such a climb because very often this kind of test being performed with non-purified bacteriocins or performed with the semi-purified bacteriocins. And we know very well that the antimicrobial activity of some of the bacteriocins can have the synergistic effect, synergistic interaction with some of the components of the growth media. Let's say some of the surfactants like SDS, EDTA, they can help the bacteriocins to have activity against gram-negative. So when we climb activity of the lactic acid bacteria-produced bacteriocins against gram-negative, we need to be sure that we work with the purified bacteriocin, pure protein. So only this way we can climb this. And they have some evidences that this can happen. Some paper was reporting on this and showing that activity of the lactic acid bacteria-produced bacteriocins can be very effective against viruses, Mycobacterium, yeast, fungus, gram-negative, including Escherichia coli, etc. But this needs to be associated with a proprietary test. Why the bacteriocins are so important? Because actually they can be maybe helping to escape this problem of antibiotic resistance. If we think about the last, let's say, 20 years, almost no new antibiotics coming on the market because it's a very simple. A pharmaceutical industry don't want to invest too much in the new antibiotics because you invest, you produce new antibiotic, you generate a new probiotic. This could be modification of existing. This could be completely new antibiotics. But then coming the antibiotic resistance, and in a couple of years, you need to look for a new one. So you're spending a lot of money for the product that could be on the market for the very short time. So it's a kind of, I call this a devil's circle, because we develop new antibiotics, and in the short time of period, we need to look for new antibiotics because already this one is not working because of generating antibiotic resistance. Or sometimes I like to joke that the bacteria, but especially pathogenic bacteria, are not stupid. When we develop new antibiotic, they found a way to defend against this antibiotic, so we need to look for another one. So maybe bacteriocins will be a way to help to go out of this circle because they work in the completely new approach. It's interesting to mention that the bacteriocin using normally the receptors that are located on the cells, and they're compulsory for the cell life. I mean, lipid 2 used there as a receptor for the niacin, as well for the vancomycin. It's a receptor that we cannot remove. Bacteria need this receptor for the another purposes. So bacteriocin actually using a very clear strategic. They attacking the test organisms, the pathogens, via the very simple receptors, and in this way they can actually act with them. It's interesting to mention that some of the bacteriocins, when they applied in the high concentration, they can use normal channels in the cells, cell walls, and in this way they can penetrate the cells and do their jobs. Some of the bacteriocins produced by the gram-negative, not a gram-positive, by gram-negative, they actually can go inside the cells by the normal channels in the cell surface, and then make a contact with the ribosomes, make a contact with the DNA, RNA, and once they do this contact, they actually interfere with the normal machinery of reproduction of the cells. Imagine if you block the ribosomes, obviously you will block the protein synthesis and cell will be dead. There was a presentation in the earlier this week of the colleague from the US. He was showing all these mechanisms when the bacteriocins from gram-negative actually using the different part of the receptors on the cell membrane associated proteins to way to block the life cycle of these cells. So it's a very well mapped from the gram-negative, but as well from the gram-positive, we know quite well the mechanisms, how they work, and how they're able to interact with the cells. So in this today presentation, we're going to discuss some of the mode of action, safety, cytotoxicity, delivery system, medical applications. But let's go with the classification of the bacteriocin from the lactic acid bacteria, from the gram-positive. There are several classifications in the literature. Very often, the new classification popping up like mushrooms after the spring rain, because very simple reasons. Some of the authors just looking for the citation of their work to increase their H factor. But there is some classification very well established from the very well-known research groups. And I really have admired the classification proposed by the John Tack, one of the, let's say, I call him Papa of the bacteriocins, because he's really person that done such a enormous work in the area of the bacteriocins. And he was classifying the bacteriocin in a very simple way. Class 1, antibiotics, this bacteriocin with modified amino acids in their structure, lanthinoin and beta-methyllanthinoin. And a typical example of this bacteriocin is niacin. And it's one of the oldest well-known bacteriocin produced by Lactococcus lactis, and actually with some other groups of lactic acid bacteria. Then we have class 2 bacteriocins. They are smaller than 10 kilodalton. That is important to mention that they are smaller than 10 kilodalton, because this small size, in fact, is important for their behavior. Since they are very small, in fact, they are polypeptides. Some people even call them small proteins, but in fact, they are polypeptides. And this small structure makes them much more stable in the high temperature. Typical examples of this is pediocin and pediocin-like bacteriocins, and they having a very conservative region in their protein structure. In this way, they're very easy to work with them, because we can target this protein structure by the appropriate biomolecular tools, and we can detect the gene sequence responsible for this protein structure, and easily to detect them in the genetic material, and to be working and to characterize them, even to purification and all this process associated to the study of the bacteriocins. But they're interesting that almost 100% of them, almost all, they're having activity against Listeria. So if you want to reduce Listeria, anti-listerial proteins, pediocin-like bacteriocins, maybe it's a good answer. And they can be produced by name, you can always guess that is Pediococcus, much more unknown bacteriocin producer of pediocin-like bacteriocins, but as well several other group of lactic acid bacteria can be producer of pediocin-like antimicrobials. Then we have class 3, that is larger than 10 kilodalton, and this is actually a small, let's say, challenge to study them, because since they're bigger as a molecule, they having much more complex structure, and this make them a little bit complicated from the point of view that could be not always thermostable. And in this way, this will get a problem if you want to apply them as let's say, as a sterile solution for application, because we need to look for the appropriate way how we can actually be sure that we're applying bacteriocin, we don't apply another antimicrobial. Small like a pediocin or niacin, we can even autoclave them, and they will be completely stable after this thermal temperature, most of them. But this, the group 3, if you put them in the high temperature, most probably will be deactivate them. And then it's coming to the class 4 of the bacteriocins. This was actually circle peptides. And now it's coming a problem, how we can do the sequencing of these proteins, a little bit more complicated. Not possible, but yet a little bit more complex way of study. One of them, enterocin SA48, is one of the actually book examples of the circle bacteriocin, been extremely well studied by a group of Antonio Galvez in Spain, and several applications of this bacteriocin been proposed, including food biopreservation and even some medical application for this specific bacteriocin. But in the last 20 years, some more examples of the bacteriocin with the circle structure is going out in the literature. So how the bacteriocins work? We need to start maybe with a very good citation from the ancient Greek Hippocrates. He said that the food will be a medicine and medicine be a food. In fact, this citation is wrong. He was never say this, and this was kind of generated in the several articles, and repeating things that is not really true become a true. In fact, he was telling that the slightly difference, he was talking about that a property food can help the medical applications of the curing the disease, but not the food will be a medicine, medicine be a food. So this is maybe a thing that we need to look in the historical book and see how we can interpret this. But there's some articles already criticizing this citation because they're providing some interesting arguments to show that it's not such a true. But it's actually a nice slogan that it's always giving a force to think that with the property of lactic acid bacteria, we can actually achieve the way that food products can help in our health status. So let's go a little bit further. We can use one interesting product produced by Balkan Peninsula's community. It's called buza. This fermented food product is made by fermentation of cereals, and it's a drink. And it's having a very long history of this product, showing that having benefits for the several medical cases. Well, we always can look on the traditional food products and see how the traditional medicine recommend them to be applied in the different medical cases. And then we can see what exactly it's a component, what exactly the metabolites are important in this product, so could be isolated and then use them as pharmaceutical compounds. This product is very rich on the vitamins because it's rich on the yeast and lactic acid bacteria. And yeast are very well known that some of them could be a producer of vitamins. There was interesting thing that slightly alcohol, but very low level, so it's this was the reason was banned for some cultures in the past, but some other cultures was continue using. Traditional medicines say that this product can be even applied for the not to cure, but to help to reduce the effect of the influenza virus. Even people with tuberculosis been recommended to take it. And we slightly in the time when we study these products, we actually know what's going, because metabolites produced by these microorganisms involved in the fermentation of this product actually important producer of antimicrobial substances, producing of vitamins, producing of bioactive molecules. Product is very well easily made. We have the grains, we boil them, smash, mix with the water, slightly ferment, and using. Well, in the past, people used the sugar to make this product, and this was actually enriching with the high number of lactic acid bacteria in yeast. These days, they're using sweeteners, but you know the bacteria don't like sweeteners; they want a real sugar. So, in fact, we might be most probably using sweeteners. We're going to lose some of the beneficial microorganisms in this product. So when we talk about fermented food products, traditional fermented food products, we need to keep in mind the traditional way of production. Modification, reducing some of the components, reducing some of the sugars, reducing some of the processes, modifying process. This can affect, in fact, on the microbial population and production of bioactive compounds, production of the bioactive peptides, production of the antimicrobials, production of the vitamins, and we, in fact, will change the product. So traditional is traditional. New products is new products, is different thing. When we're talking about the effect of the modification of these microorganisms, it's interesting that we observed in case of this one of the strains of in this case, Bacteriocin ST69, produced by Lactobacillus plantarum, isolated from buza, was showing that we can measure percentage of the cell lysis of Listeria, of Enterococcus, of Lactococcus sacchi. And you can see that even small amounts of the bacteriocin was able to do the very high level of cell lysis of this test organism. So it means the bacteriocins are much more, they're effective even in the very small amount of the presence. In nanomolar concentration. And they can be effectively inhibit the growth of the food spoilage and the food pathogens. Same time, we was using the atomic force microscopy to observe the effect of this bacteriocin, same one produced by Lactobacillus plantarum, and we was observing then when we apply the bacteriocin to the cells of the target organism, we actually doing a pore formation. And the pore formation is well-known mechanism of action of antimicrobial peptides, bacteriocins, against test microorganisms, especially this from lactic acid bacteria, because they attach to the receptor or to the specific target of the cell surface. They do the pore. Once they do the pore, the bacteria will lose the intracellular material and they will reflect to the death of the cells. And the good point of the using atomic force microscopy, it's a fact that you work with the live cells. If you look with the scanning electron microscopy or other type of electronic microscopy, you need to kill the cells first. You need to coat them with gold or with platinum and to see what's going, because it's interaction of the electrons with the cell surface, in fact with the metal, put it on the cell surface. But with atomic force microscopy, you have the real life cells. And even if you have a patient to take a several picture one after one, you can actually see what's the dynamic of the process, how the cell is destroyed and how they're losing intracellular material. Another way to see this interaction is to look for some losing of some metabolites. Think about the beta-galactosidase. It's an enzyme that is intracellular enzyme. But if you destroy the cells, you're going to detect the beta-galactosidase in the extracellular environment. So beta-galactosidase, it's a very good enzyme because he can transform a product, ONPG, to the substrate to the product. So you have substrate ONPG, you can transform to the product, and the product have the color. So you can do the treatment of the cell suspension with the beta-galactosidase, and you will change the color because you transform a specific product for the enzyme to the substrate for the enzyme to the product. And if you provide this substrate to the this cell surface of the test organism, you put the substrate and you detect the change of the color. It means enzyme was present because he do the enzymatic reaction. And this is the good way to see if the cells are destroyed. And if you have the bacteriocin that destroys cells, you will detect the enzymatic reaction by see the change in the color. The different ways to detect the change on the non-treated and treated cells in the RNA and protein concentration. Again, this is quite a simple way using the spectrophotometrical approaches, and you can actually detect these changes. So you can see that the bacteriocin, in fact, working. In the previous presentation, I mentioned about the antiviral activity for some of the bacteriocin produced by Enterococcus munti. And in this case, we have the activity of this bacteriocin produced by the strain isolated from Bosah. All of them are lactobacillus, formal lactobacillus, so now we need to call them lactobacilli. And then there was having some effect on the viruses. As well, all these tests needs to be performed together with the fact that we need to be sure what is cytotoxicity. Because cytotoxicity of the bacteriocin is a very important point, because this can be related with a way that you can apply. Every bacteriocin is a different protein. We need to know if this specific protein have this or not have cytotoxic effect. Some of these strains isolated from Bosah was producing component that was having a very good activity against Mycobacterium tuberculosum. And this was actually explaining, we have the control. You can see that our strains was not reaching the control, but they was having some very good high activity in the inhibition of Mycobacterium. And this is a good point because we can develop probiotic product with activity against Mycobacterium. Again, not going to cure the disease, and you don't want. Probiotic not supposed to be curing the disease, he needs to help. So if we develop a probiotic that can be given to the target population that be can be exposed to the Mycobacterium tuberculosum, to the tuberculosis disease, this can maybe protect them of the developed disease, or can help to the customers, to the patients that already have the disease in their cure process. And this explaining why traditional medicine was recommending this drink for the patients with the tuberculosis, because they're having these antimicrobials inside the product, and at the same time reaching the vitamins actually boost the immune system of the patients and helping them in the natural defense against tuberculosis. When we talk about the application as well, the mode of action, it's interesting to mention the fact that some of the bacteriocins could be having synergistic effect when they are applied with the antibiotics. And in this case, we're having some of the effect on the synergism when we apply the low concentration of antibiotics together with some of our bacteriocins produced by the strains isolated from Boza. Now, how we can apply some of these bacteriocins in the human and veterinary practice? If we see this presentation, it's from one of the papers of Paul Cotter. He was summarizing very well the different area of application of the bacteriocins. They can be applied in the food quality, because if you have the starter culture that's producing antimicrobials, we can guarantee much more unification of the microbial population of the fermented food products, because these antimicrobials, they will take care to reduce the level of the other microorganism in the food product. So the food quality, we want to produce the food with the same quality, not with the surprise presence of the different microorganisms inside these products. Food safety, antimicrobials as a bacteriocin, example, or strain-producing bacteriocin can be having a very good effect on the reduction of some specific target pathogens. So they can even reduce presence of listeria. Niacin is very well applied for the reduction of staphylococcal and listeria contamination in the several cheese products. Well, some of the cheese products that you can buy here on the in Brazil on the not legal market, like homemade or the people that's selling their homemade products without authorization, sometimes you can observe this small gas formation inside the product, like a small balls inside the product, is showing that there is some contamination. Very often this could be enterococcal contamination, and obviously we're not going to expect the bacteriocin from lactic acid bacteria to be answered to reduce this contamination, because Escherichia coli enterococcal contamination are gram-negative. Well, bacteriocins from gram-positive, from lactic acid bacteria, not supposed to be. Some of them could, but in general not. But we can look for always the bacteriocin from gram-negative, safe microorganism that can use to control the gram-negative contamination in cheese products. From the veterinary medicine, applications of the bacteriocins fact that they use for the, let's say, for the mastitis in cow. They're using already in the human medicine modification of some bacteriocins, and even natural produced bacteriocins for treatment of middle ear infections, for treatment of tuberculosis, for treatment of other multi-drug resistant Staphylococcus aureus, as an example, on skin infections. So there is more and more examples. And very often, we need to think about the options that we need to modify the bacteriocin in order to improve their quality and their mode of action, so it can be more effective and applied in the different products. There is an example of the application of the bacteriocin produced by Streptococcus macedonicus. And this strain was isolated, interestingly isolated from yogurt produced by the Bulgarian company working in South Africa, and most probably having some relation with the strains that have been isolated in Greece and Macedonia, because of fact that it was isolated from the dairy products there. But since the company is from Bulgaria, most probably they was using starter culture from the Balkan Peninsula. But so these days we don't know anymore from where bacteria is coming, because there's interesting examples, Streptococcus infantarius. isolated in northeast of Brazil. But this strain was associated with the dairy products in Africa. Maybe during the like 200, 300, 400 years ago, when the slaves been moved from the Africa to Brazil, maybe they already transport these strains with their fermented products, and this is why the strain actually been isolated from northeast of Brazil. So humans actually transporting all these microorganisms, even with the strict regulation to don't transport food products. We always have these strains in our gut system, and humans could be a factor that can transmit the microorganism. Just think about all the disease that been brought by the Spanish and Portuguese in the Latin America and kill almost the entire indigenous population in the past. But by the way, there's interesting fact that some of the Maya people in the Mexico, they've been killed by salmonella that been brought by the Spanish people conquisting this area. Interestingly, this one that was the sender of this native people, they having resistance to salmonella, because obviously there was small part of the population that was having resistance to salmonella infection, and only they was able to survive this introducing of new diseases. But interesting that this strain of Streptococcus macedonicus was having a very good effect against some of the mastitis-associated pathogens in the dairy production. We was observing this by growing the test microorganisms, Streptococcus agalactiae, associated with the mastitis, by just growing and observing how the culture grow when it's presence of the bacteriocins. Or we was showing the same thing with the atomic force microscopy, showing how the cell losing intercellular material when they was exposed to the presence of this bacteriocins. Or even we try to generate a kind of product that can be applied in the animals and showing that they can release the bacteriocins and kill the test microorganism. But we wanted to go a little bit further. We developed actually a pool, several 6 test organisms we put together to grow together, and in the presence of this bacteriocin producer, Streptococcus macedonicus. And we was having a different culture, as a single culture, like 1 to 7. 1 is the bacteriocin producer, 2 to 7 are different pathogens associated with mastitis. And the 8 is all of them together. So we do the DGG analysis, and we wanted to see which of them is present. Obviously, you can see that some of them, they cannot survive when you have the mixed culture. So they grow together with bacteriocin producer. Let's say the strain number 6, the band for the strain number 6 as a single culture, it's present in the single culture, but in the mixture it's not. Number 1 is a control. It's our bacteriocin producer. So he's present in the mix. The number 3, it's present as well, but number 2, it's not. Number 4, it's not. So you can see that when we grow in the mixed culture, we can have an effect of the bacteriocin producer eliminating some of the other organisms, all of them associated with the mastitis. Obviously, you cannot eliminate everything, because number 3 was quite successful and he continued to be in the mixed culture. So again, we don't have panacea, we don't have something that can eliminate everything, but we can help. The problem is that mastitis microorganisms, some of them can survive pasteurization, and they can be present in the dairy product. And they can interfere with the quality of dairy product, and they can even interfere, and some of them can be even a problem for the customers, because when you have dairy product contaminated with some of the mastitis-related microorganisms, this can maybe give a problem for the consumer, especially small children, very old people, some of the immunocompromised people. And now it's coming how the bacteriocin in fact work. We saw in the other presentation some of the mode of action of bacteriocins from gram-negative, but gram-positive, they work slightly different. Some of them, very simply, they can go and install them inside the cell wall and destroying the formation of the cell wall. In this way, they making microorganisms more vulnerable for the destruction, like a group of Bacteriocins, the last one. In the middle, you have the nisin that coming and recognizing lipid 2. And once recognizing lipid 2 by the N terminal, the Z terminal actually going inside the membrane and doing the pore formation process that will result in the destruction of the test organisms. Or like a saccharine, like a class 2 Bacteriocins, they can go install them in the cell membrane. And then once they install them inside the cell membrane, because of the normal interaction between membrane and the protein, they can actually interfere as well with the normal biological function of the test microorganisms. So, but other way how they can interact, some of them can use the different receptors, not only lipid 2. They can several receptors they can use. The mannose transporter can be used for this process. Other proteins, enzyme, proteins regulator, etc., can make a contact with the Bacteriocins, and in this way they can interact with the normal machinery of the microorganism. Some especially gram-negative, they can go inside the cell and then interact with the RNA and DNA, and in this way they can interfere with the normal reproduction of the normal processes. So proteins with different primary role but not expressing antimicrobial activity, it's another point. In the beginning, we were thinking that if we detect antimicrobial activity, this is a protein that it's there to kill, not always. Because Mike Chikindas have this that I presented in the first day, that some of them actually are quorum-sensing metabolites. By chance, they having antimicrobial activity. Their primary role is not to kill. Their primary role is to interfere in the communication. They having a completely different structure. There was shown that some Bacteriocins are like histone-like proteins, and histone, we know that are important in the structure of the nucleus. But by any chance, these proteins showing antimicrobial activity as well. And it's very interesting when we do the antimicrobial activity, all these different proteins, they have a completely different role, and by chance they have antimicrobial activity. Normally they don't have high activity level, they have very low activity. So it's very interesting because we can see that Bacteriocin is much more complex world, and we need to actually evaluate and see if this protein that we detected that have antimicrobial activity, in fact, is really only antimicrobial, maybe having completely different role. And in this way, we can actually giving much more better answer what's going in the life system. When you're talking about spectrum of activity, we need to always think about that by definition, Bacteriocins are active against closely related. So we need to know what of the closely related microorganisms we can inhibit. But at the same time, we need to know how we can interact, if they can kill or not, some important food and human pathogens, because we don't want food-related associated pathogens. We don't want medical associated pathogens, but as well we don't want to kill the starter culture or other beneficial microorganisms in the system. So if we apply let's say Bacteriocin or Bacteriocin producer in the preservation of cheese, you want to kill Listeria, because Listeria we don't want, depends, we don't want it in general. But in the same time, we don't want to kill starter culture. Because if you kill the starter culture, you destroy the product. So it don't make sense. You make a yogurt. You can add the Bacteriocin to control the spoilage pathogens related to mastitis. But you don't want to kill Streptococcus thermophilus and Lactobacillus bulgaricus. So obviously we need to have this answer. We don't want to kill the probiotic bacteria, because very often we can add the probiotic bacteria in the dairy products to deliver this probiotic to the customers. But in the same time, if a Bacteriocin producer kill the probiotic bacteria, what's the point? You have the very safe product, but it's not anymore probiotic. So all this needs to be tested. And as well, the evidence is showing that some unusual activity could be expected. We can observe very interesting activities. So let's check, let's see, gram-negative, if we can kill it, but please use the same purified Bacteriocins, because test first with not purified, but if you observe something, go and see with the purified Bacteriocin if this is a really activity of the Bacteriocin, or it's a combined effect between Bacteriocin and something else. see if we can kill some viruses, if we can kill Mycobacterium yeast fungi, because this will give it more straight to our bacteriocin and see how we can apply it and use it. Some examples, some of the bacteriocins produced, especially this one by Enterococcus munti, have this nice activity against some viruses and showing some potential applications. We're having this explanation why these bacteriocins can have activity against some viruses, and work performed by Monica Washman from University of Buenos Aires was showing that one of the proteins, gamma proteins, by the virus cannot be synthesized when the bacteriocin is present. Very elegant and simple work, but a very elegant way showing the mechanism of the presence of the bacteriocins and how interfering with the reproduction of the viruses. Well, some of the other works was showing that some other viruses can be inhibited by some bacteriocins. And it's very interesting point. Most of this work by enterocins. Why? I have no answer. Most of the work was showing this, and some of them have done by as well by Monica and colleague from the US. We was watching yesterday presentation of Mike Chikindas. One of his bacillus bacteriocins was having as well some interesting effect against some viruses. But why not so much activity of the other group of the bacteriocins? Something that maybe deserves more attention and better study and understand what's going. Then we have the other bacteriocins and antiviral activity. So we have this CR35 Enterococcus faecium that later been reclassified as Enterococcus munti. The multiplication of the virus, it's what's going, what concentration of this bacteriocin is needed. Some other bacteriocins and antimicrobial peptides like labirinopeptide showing the activity against human simplest virus. Some of the viruses even showing the activity against even the HIV virus. But then it's coming a problem that very often this is associated with a very high cytotoxicity. We know that some cases this could be proteinase inhibitors. So this is very important. All this work needs to be done with the purified and really sure that we're using the bacteriocin and don't using something else together with the bacteriocin. Some of the application of clinically significant of methicillin resistant Staphylococcus aureus could be inhibited by some bacteriocins and showing great potential. What we have more coming to the Mycobacterium tuberculosis again, several bacteriocins can show the activity against Mycobacterium that is not typical for the bacteriocins, but we know that some of them can work. Colleagues from Ireland, they was showing even how we can modify the antimicrobial peptides and to improve this activity. So there was actually making modification of some of the amino acids in the structure of the bacteriocins by the chemical replacement or by the modification, genetic modification, and actually forcing the microorganism to produce the modified bacteriocins. And in this way, they can improve the activity against Mycobacterium. Candida albicans, again, not supposed to be targeted by the bacteriocins, but we can see that live culture and even the bacteriocins can have the activity against Candida albicans. And this is important because this is a microorganism associated with vaginal candidosis. So it's interesting to see how actually this bacteriocin produced by Enterococcus munti CL35, strain property of Cerella in Argentina, and was actually interfering with Candida albicans, and a very small amount of microorganism was able actually to do the destruction of Candida albicans. So we wanted to see in this case how we can this process of the absorption of the bacteriocin to the Candida albicans can be modulated by the temperature and the pH. Why the temperature? Because obviously body temperature is 27. But we want to know lower and higher temperature, how they interact, because to compare, to know if the 37 is a really a good temperature. And as well, how the pH interfering with all these processes, because when you have the normal vaginal environment, it's a slightly acid. But when you have the yeast infection, pH go higher. So obviously you want to have it in the high pH because this is the way when you have the infection, and as well in 37 because this is a body temperature. And then if you add the constant contraceptive, how all this environment will be affected. And it's interesting to observe that aggregation not only between Bacteriocin, but now between Bacteriocin producer and Candida albicans can be actually interfere by the presence not only on the pH, but on the presence of the contraceptives. And you see when you change the pH, how it's actually aggregation between the Bacteriocin producer and Candida is changed, because this is at 3, you have it at 5, and you have it at 7. So idea, if we can use this microorganism, and yesterday Professor Suvorov was really defending importance of enterococci, because they're not such a bad guy. Several of them are very beneficial microorganisms, and they're part of the several fermented food products. Enterococci actually can be a good, well, some of them could be really bad because of the antibiotic and virulence factor, antibiotic resistant virulence factor, but we need to check and prove that they are really, this one strain that we're using, it's a really safe, can be used. But again, Enterococcus munti could be applied as a probiotic for control of the Candida albicans in the infection process, and because can be really effective on the elimination of Candida albicans, because producing something that can kill Candida albicans, and as well can interact very well with this microorganism. Then we have control of metallicin resistance to Staphylococcus aureus, several Bacteriocins, antipidermin, gallidermin, mutacin, Bacteriocins from the different groups of microorganisms, and they will be able to help in the control of metallicin resistance to Staphylococcus aureus, and they're a big problem in the medical settings. Lactacin, different lactacins, and showing effect by different groups, they can be as well effective. Salvaricin A and B, Salvaricin A produced by Streptococcus salivarius was having good effect against Streptococcus purulens. And Streptococcus purulens is a very important infection associated with serious clinical cases. Bacterial vaginosis as well can be used, different Bacteriocins in the control of bacterial infections in the vaginal environment, and they could be associated with the production of the antimicrobial proteins by some lactic acid bacteria. So probiotics and the Bacteriocins could be an option for this treatment. Bacteriocin in the application of the biofilm prevention. We can prevent formation of the biofilm. We can destroy the biofilms using the Bacteriocins or using Bacteriocin producer. Antifungal activity as well, several works done in this area showing that the Bacteriocins could be as well effective against some fungus. How we can deliver this Bacteriocins? We can use the protein capsule, we can use the liposomes, nanofibers, incorporation on packaging material, cyclodextrins, different silicates, dendrimers, nanoemulsion, nanogel, solid lipid nanoparticles, etc., etc. And this is a different options, and important when we incorporate the Bacteriocin in certain delivery system, we need to have a slowly release. Because if you add the Bacteriocin not incorporated in nothing, we have immediate effect. But if you can incorporate in some vector delivery system, we can liberate the Bacteriocin in a long time period, and we can have it a better effect and for the long time. Even in formation of nanofiber, quite simple method, we can even design this in the lab and perform it, is showing that we can incorporate the Bacteriocin in the nanofiber and they will be delivered for the long time. Even we can modify the Bacteriocin. We can do the application of some copolymers with the Bacteriocin. And in this way, we can improve their resistance to the proteolytic enzymes, because the biggest problem with the Bacteriocin, they are polypeptide. And if you have the proteolytic enzyme, they will destroy them. But we can incorporate some of the polymer on the N-terminal, and in this way, we can actually improve their stability, and we can have them better. Why it's not moving? Okay, because its time is running, we need to say something about cytotoxicity. Everything in the pharmaceutical industry, in the food, is toxic. Just the question of the quantity. And interestingly, the bacteriocins are quite considered as a not toxic. But again, it's coming a problem. What kind of cell lines we need to apply to check this cytotoxicity? It's important to use the standardized procedure, and at the moment, we don't have really well-established model. Most probably, we can use cell lines recommended by pharmaceutical industry, because we're going to move in the next decades from the just clear food application to the pharmaceutical application, and maybe we need to adapt to the pharmaceutical standards and use the specific cell lines for the detection of cytotoxicity of the bacteriocins, to be sure. We know that the niacin is not cytotoxic because he has similar cytotoxicity to the sodium chloride, almost 7 grams per kilo. That is considered a very good low cytotoxicity, so it's not cytotoxic. But again, every bacteriocin is a different protein structure, so obviously we cannot generalize, and just because niacin is not cytotoxic, nothing else it is, because we know that some of the bacteriocins in fact can be cytotoxic against some cells. And since very interestingly, using very often cancer cell lines to check the cytotoxicity, and high cytotoxicity against cancer cells, it's a very good point. But because if the same bacteriocin don't have cytotoxicity against healthy cells, it's a very good application. But all this needs to be evaluated and checked. And Cow, they suggested that several bacteriocins, in fact, could be very interesting to be applied as highly cytotoxic against cancer cell lines. But again, all this needs to be evaluated and checked if their cytotoxicity is not against healthy cell lines. And even we can suggest that some of these bacteriocins can be applied in the control of the cancer cell lines. So in fact, this is a good point, because highly cytotoxicity to the cancer cell lines could be maybe a way of treatment of some type of cancer. And for the finishing, I want to just remind you again that we're going to have this IFP Latin America conference in November, 11 to 14 of November. Deadline for the submission of the abstract is still end of this month, so we still have like 5-6 days to submit your abstract. And it will be interesting to participate in this meeting that will share information about the food safety issues in the all Latin American world. Presenters you can check on the website from all around the world, and as well the Yakut conference on the innovative application of fundamental science in the application of the lactic acid bacteria and other microorganisms and the antimicrobial peptides for the good of the food and health. It will be next year in March in São Paulo. And again, for the finish, don't afraid to be innovative, as I already said before, and creative, because Ark of Noah been built by one person, and Costa Concordia and Titanic by the professionals. But one survived, and two, it's on the bottom of the sea. And thank you very much for all my collaborators from all around the world, because work, it's only possible if we collaborate with our colleagues from different countries, and in this way we can go in the front and achieve our goal. Thank you very much for the attention, and now questions. Any questions from the, maybe, yeah, okay. What's the usual booze alcohol content more precisely? It's less than 1%. The actual MAPA beverage registration required that not alcoholic beverage less than 5. Okay, well, it's less than 1%, depends of the time of the fermentation. And again, this drink, it's very well accepted in the Balkan Peninsula. I already tried to give this product to some colleagues in Europe, South Africa, Brazil, not very well accepted because it's specific taste. But again, if we can control the fermentation process, and even if local regulations say that needs to be below 0.5, there's no problem, because yeast fermentation can be controlled and can be lower, and we can count only on the slight yeast fermentation and predominant of lactic acid bacteria. Only we need to see if the Brazilians will like the product, because it's a really specific taste. By the way, no, I'm not going for these examples because maybe they will. Well, traditional medicine giving this product for several applications. Even there was like traditional medicine say the woman that has the problem with lactation, she can drink this drink and will improve the way she can produce the milk. And the reason it's very rich on the yeast. This is actually helping, not because of some magic. There's too many yeast inside and lactic acid bacteria, vitamins, all these kind of things. It's a complex process and it can help. Not replaced, because if there is no production, it's no, but can help. We have the questions here from the audience.
Nice presentation, Professor. I really enjoy it, so thank you. How the bacteriocin producers and other acid-like bacteria are immune for their own bacteriocins?
Well, bacteriocins producer, they having their way to be immune to their own bacteriocins. So you don't want to kill yourself. So when they're producing the bacteriocins, the bacteriocins actually can interact with the receptor, and they was stimulating autoimmune response. If you can call immune response in the bacteria, but they do. In the same time, they having receptors, they can catch the signals from the other molecules, and they can do by the dephosphorylation, and they can activating expression of the bacteriocin producing operon. So this could be their own bacteriocin can serve as an inductor. So one bacteria actually can give the signal to the other bacteria to start producing, or could be even surface proteins from the competitors. So listeria, some listeria surface proteins can serve as an inductor in this dephosphorylation of the ATP to ADP, and then activate the operon expression. So once the bacteria discover that listeria is around, because of detecting this surface protein, they can actually start producing the bacteriocins. So in some cases, this process can be induced, and it makes sense because it's a competitor for the food we're going to produce, and we're going to eliminate these competitors. But at the same time, they're producing these signal molecules that acting to induce the immune response. So you don't want to kill yourself. You want to kill only the intruders, this one that want to compete with you for your food. This is why they kill the closely related, because they're having required for the more or less the same ecological space and source. Yes. Can you pass the microphone, please?
Well, congratulations, Professor, for the wonderful presentation. Please, Professor, my question here is there any challenge of using maybe any bacteria strain in nanoparticle or nanoemulsion?
Well, in the agriculture, we're having already some bacteriocins for the treatment of mastitis. There's a commercial products, and as well we're using several of the probiotics. They are producer of the antimicrobial peptides and used in the poultry and animal production to reduce the infection and some of the pathogens associated with this with the production of these animals. They are. And we need to move to the next presenters because it's already 10. There will be no break for the coffee. Next presenter going to be by a video presentation. It's a Professor Iskra Ivanova from Sofia University. It's interesting to mention for the CV of Professor Ivanova that she was graduated from Moscow State University and a master and PhD from Moscow State University. She was working in one of the pioneer companies later in Bulgaria on the development of the biogenic stimulators, and then become member of the Sofia University research and professor body. She was involved in the several European projects and lecturing in the different European countries. A few years ago, she was invited professor in Brazil for almost 3 years, working in University of Viçosa and University of São Paulo. So it's a really big pleasure to have Professor Ivanova presentation today, and she's going to talk about some bioactive peptides produced by the lactic acid bacteria and fermentation of the dairy products.
Ivanova, and I am professor from Sofia University. Lactic acid bacteria are bacteria which contribute to the diet of mankind. I would like to pay the attention on the 3 photos on this side. The first one is famous French scientist Louis Pasteur, who discovered the principles of vaccination, microbial fermentation, and pasteurization. The second photo is Ilya Mechnikov, a Russian scientist, Nobel Prize, who developed a theory that aging is caused by toxic bacteria in the gut and that lactic bacteria could prolong life. The third photo is the photo of Bulgarian scientists. For us, Bulgarian is a very important man, and because he made a wonderful discovery. He discovered that a certain strain of lactobacillus is the true cause for the existence of natural yogurt. In recognition, the strain was called by the scientific community Lactobacillus bulgaricus. His name is Stamen Grigorov. Dear colleagues, again, I pay your attention about lactic acid bacteria as cell factories. And the concept of lactic acid bacteria is as a group of microorganisms developed in the beginning of the 19th. The interaction of lactic acid bacteria in foods enjoy early attention of scientists and resulted in significant contribution by Pasteur on lactic acid fermentation in 1857 and the first isolation of pure bacterial culture, Bacterium lactic, by Lister in 1873. Lactic acid bacteria are typically involved in a number of spontaneous food fermentation, but they are also closely associated with human environment. Maybe it's interesting for you to know from where comes the word yogurt. And in the next slide, I will continue speech about some history of these milk products. The word yogurt is composed one as is of Han and Altai derivation. It consists of yolk, which means fat, thick, and turt, milk. As I promise you, I will tell you something about the history of milk products. Regarding a period 7,000 years before Christ, were discovered vessels for collecting milk and that bear evidence for early utilization of animals for milk production. The excavation work in many countries confirmed that such fermentation were used over 5,000 years ago. An example, for instance, for sourdough bread was part of the typical diet. The living of sourdough bread was mentioned in the Bible in Matthew 13:33. I continue with the history, but on this slide, I would like mostly to focus what was found in the lands of Bulgaria and with what history is connected to these milk products. I will speak a little about Thracian tribes who also contribute to the history of milk products. Information about Thracian tribes as good farmers and stock breeders who kept large sheep flocks and found a natural way of obtaining lactic acid bacteria, as for example yogurt. Herodot is the first author to report yogurt as a gift from Thracian. This also was confirmed by Hellenic geographer Strabon. Virgil in it makes the following remarks about Thracians. They drink yogurt, blending it with horse blood. Obtaining yogurt as a product of its own was determined by the period of the start of domestication of milk elements, as well by the time required for the accumulation of experience and production traditions. Dear students, it was interesting to speak about history, but now we have to pay attention on more serious slides. And this presentation now will be focused on proteolytic enzyme system in dairy lactic acid bacteria. This is a very difficult thing to understand everything, because the study of proteolytic enzyme system of any bacteria is connected with a lot of knowledge in biochemistry. And now I will try you very briefly to speak about this system. Proteolytic system of lab converts proteins to peptides and them to amino acids, which are essential for bacterial growth and also contributes significantly to generation of flavor compounds as end products. The optimal lab grown in milk, it relies on sophisticated proteolytic system that includes a cell wall associated proteinases. You can see here they are encrypted like PRTP, several membrane-located peptide transport system, and the array of intracellular peptides performs the first step in the degradation of casein, yielding peptides in the range in size from 4 to 30 amino acids. DTPT is a secondary transporter that catalyzes the uptake of hydrophilic D and tripeptides. OPP and DPP systems are members of ABC, this means ATP binding consent transporter family, and are composed of one of OPPA and two OPPA and DPPP oligopeptide binding proteins and membrane-bound complex formed by two hydrophobic integral membrane proteins and two membrane-associated proteins that carry the ATP binding cassette motif. The oligopeptide binding proteins determine the specificity of the transport system and therefore the range of the peptides that may enter the cell. OPPA transport peptides ranging in the size from 4 to 35 amino acids. DPPA is a specific for D and tripeptides, whereas the DPPP transporters peptides up to at least 9 residues in length. Casein-derived peptides from 4 to 30 amino acids in length produce PRTP can develop be only transported by another systems. Once inside the cell, oligopeptides are further processed by the intracellular peptidases, yielding finally free amino acids. Now, on this slide is in 4 points. I tried to pay your attention about what is this protein hydrolyzed bioactive and how they are found in nutraceutical and functional foods. First, protein hydrolysates or bioactive peptides have been consumed in the foods for a very long time, and now scientists focus their efforts to identify specific peptides and their role in the nutrition. At the same time, manufacturers refine their process and embrace it with the latest technologies to develop specific protein hydrolysates bioactive peptides. Further, the scientific evidence is also increasing to use them as nutraceuticals. I will say later what is nutraceutical and functional foods, remember. And as a result, this field is rapidly growing and sales of protein hydrolysates are increasing year over year. On this slide, we will speak about a little about structure and size. You can imagine that peptides and their number is enormous, but exists some knowledge about what is the structure and what is the size of the peptides. Of course, they are based on their inherent amino acid composition and sequence. This is very important. We will continue further in this lecture about what is the sequence of some very important peptides. What is the size of the active peptides? As you hear 2 slides ago, we were speaking about different number of peptides, how they are formed. And now I will tell you that active sequences of peptides, they vary from 2 to 20 amino acid residues, and many peptides are known to have also multifunctional properties. Peptides from the sequence, for instance, 60, 70 of beta-casein show immunostimulatory, opioid, angiotensin, 1-converting enzyme inhibitory activities. And important is what is sequence, where to find this sequence. And for instance, in beta-casein, exist, and in the other caseins, so-called strategic zone. This is work which are carried on, and for different proteins to find the strategic zone and from where the peptides can be easily produced is a lot of information exists in the literature. The sequence is protected by proteolysis because of its high hydrophobicity and the presence of proline residues. Here, this sentence, what is the meaning of this postulation? It is that the cell also take part of protection of the peptides. Functionality of milk proteins derived bioactive peptides and their potential health targets. You can find such pictures a lot of in the literature. I consider that this is one very good done. It's not my production, but I would like to speak about this picture. And first to pay your attention, first of weight management, heart health, bone health, mood, memory, stress control, and also immune defense, digestive health, dental health. First, let's pay some attention first on milk proteins derived bioactive peptides, which contribute, for instance, to mood memory and stress control. It's a very interesting peptides. They are called casomorphins and other opioid related. Maybe you know very well that when you were children, your mother gave you before sleep a glass of milk, and you sleep well. This was considered that it is like only the milk is important, but what is the most important, that drinking the milk were formed peptides who have such opioid action. This is a very simple example. Of course, exists a lot more about these properties of the milk, but this is the property of the milk, but the peptides were what were generated during the consumption of milk. On the other hand, milk proteins important for bone health. Here, scientists had found that calcium-binding proteins, for instance, lactoferritin and other, they're important for the health of the bones, and this is a very big problem in the contemporary life. Many people suffer from bones. That's why this is also very important that we can find something good for our health, for our back, for our knees, to feel better if we drink and eat milk products, but the peptides which are found in these milk products. The other important is heart health. You know that a lot of people are dying. This is a big problem of the mankind, the heart diseases. And here, one of the most important peptides found are so-called ACE inhibitory peptides. The other antithrombotic, anticholesterolemic. They are very important because heart and muscles are important part of humans, and they are they have a very important heart health on people, because this is the main motors. The heart is of the humans. Here, in this aspect, also is important the peptides which are antimicrobial, immunomodulatory, glucomacropeptide, and this is connected to his digestive health. Some small attention about weight management, and here safely including oligomacropeptides, which are also have opioid action. Immune defense. Immune defense is connected to these peptides which have immunomodulatory, glucomacropeptide, C2 modulatory. And the last is, but so also very important, is dental health, which are in milk products. You can find some antimicrobial calcium binding peptides, which are very important for the teeth. Now, as I told you, make a good separation to know functional foods and nutraceutical. Functional foods are consumed as part of usual diet, providing benefits beyond basic nutritional function. And the other is nutraceuticals. Are products isolated, purified from foods, provide physiological benefit or disease protection. On this slide, I can give you a general explanation about physiologically functional dairy products. And here we look the first, the left part. This is probiotic bacteria, which are part of lactic acid bacteria, and they are producers of prebiotics and also the role of symbiotics. This part is conducted by other speakers. I will not speak about this because this is far from bioactive milk components. And the bioactive peptides, they are produced from milk protein in two ways: fermentation or technology. In the case of milk proteins, bioactive peptides are generated in two main ways. The first is microbial fermentation in dairy products in a reactor, and also we can produce milk peptides in vitro by enzymatic hydrolysis. Bioactive peptides to alleviate hypertension are very important. And now I would like to give some small information what is hypertension, and this is one of the most prevalent contribution to cardiovascular diseases, which are the world's largest killers and claim more than 17 million lives per year, according to World Health Organization. Angiotensin converting enzyme, or called also ACA, plays a critical role in the regulation of blood pressure as well for the fluid and salt balance in mammals. It catalyzes the formation of angiotensin II, a strong vasoconstricting octapeptide from angiotensin I, and also inactivates bradykinin, which causes the blood vessels to dilate and hence lower blood pressure. Therefore, ACI inhibition may lead to the reduction of the blood pressure. Part of this slide, you see the IC inhibitors in fermented milk products, and is given an example of these peptides and what is included in such peptides. IC inhibitory peptides produced from caseins are found in the region 25-27 of the structure of the casein, and it is already identified and contains the following amino acids: valine, alanine, proline. IC inhibitory peptides were identified in whey proteins. Examples of these peptides are found in the zone of 142 to 148 and from 102 to 105 of the primary structure of whey. The next part of the speech is connected with our results obtained studying white brine cheese in Bulgaria. Also, I would like to note that this kind of cheese is very popular on Balkan Peninsula. It is produced from cow, goat, and sheep milk in a specific technology, and is a part of the main diet of people living in this part of the world. Here on this slide, first you see the dynamics of the free amino acids during the ripening of white brine cheese. What is important? For what I give you this picture? You, of course, from this picture, I'm sure that you will find the differences between different periods of producing this cheese and what happens with the level of amino acids. And here on the first, in the upper part of the slide, you see from goat on the first day, on the second day, the fermentation, and on the third. If you pay attention on cow cheese, you see that formation of different amino acids is quite different. That's why this is very important for the manufacturing of cheese, that amino acids vary in different cheeses produced by different mills. And for instance, it's considered that goat cheese have a high level of amino acids at the last stages of fermentation, and is very healthy for people with some health problems. Picture is about the performed HPLC analysis of samples of different white brine cheeses produced by sheep, goat, and cow milk. Peptides, you can see the beautiful picture of a cow, and what happens with this cheese and what is the amount of the peptides formed is different, quite different from the peptides formed after 30 days of obtaining the cheese. On this picture, you can see again what happens with the peptides from goat cheese and what is on the third day of ripening of this cheese. You see the differences which are remarkably different. That's why, again, I say that goat cheese is very important. It is a little different from the traditional cow and sheep milk. Sheep cheese. Look, again, a very good difference between on the first day and on the 30 day of ripening of the cheese, and different from the other cheeses from the other two milks. One of the basic nowadays analysis of peptides is using the so-called Malditov analysis. This is a mass analysis of samples, here we see, of different white branches produced by sheep, goat and cow. These pictures are not informative for presentation, but I would like to show you only the fact that these analyses contribute to the main understanding what kind of peptides are formed, and further in our presentation you will see what was done by using this analysis to know what is the difference between goat, sheep, cow, milk peptides and how they are found. Има книга, извинявай, вече. Още малко имаш, само търпение и свършвам.
Няма нищо, просто значи, прецени си на трите слайда какво да кажеш различно. Сега, това е първото, говори за шипа, после че шипа се сравнява с това и общите приказки също тук, не знам.
Общите приказки за всички са едни и същи. Malditov analysis is an important method for identification peptides. In our analysis, we use samples of different white branches produced by sheep, goat, and cow milk. On this slide are shown the amount of the peptides found by this analysis, and they also are different for this part is in connection with the time for brining of the cheeses. The same situation is the data obtained from analysis of goat cheese. Also, we have a very big differences in the analysis made by Malditov mass spectrometry, and this is, of course, confirms that our knowledge about peptides depends on the kind of the analysis done. And again, we now look cow milk, and again is the same situation that the period of ripening gives information about the quality of different peptides. On this slide, you see amino acid sequences of found peptides in white brine cheese. We take we have results from cow beta-casein, and the found peptides are found in the zone underlined. The same is for cow alpha S1 casein and alpha S2 casein. At the end of our speech, I will show you the total peptides found in different milk caseins. pay attention from one type of casein, which is called alpha S1 casein. You see that from cow we obtain 27 peptides, from goat 12 peptides, and from sheep 20 peptides. From the other kind of casein, which is called alpha S2, we found also different amount of peptides. From cow 4 peptides, from goat 4 peptides, and from sheep 4 peptides. From beta-casein were identified peptides, and again their number from cow is 20 peptides, goat 6 peptides, sheep 7 peptides. This is the last part of our presentation, and what is very important for us, like researchers of course, it is about what kind of peptides were identified. From alpha S1 casein were identified peptides which are connected with ICE peptides, casukinin, casophosphocasein, immunopeptide. These are very important peptides. They have a very important action on humans and a very important part in milk products. From the other casein, which is called alpha S2 casein, look, we find peptides mainly with antimicrobial activity. And the last casein, the very important beta-casein, we are very glad that we find a lot of enzymes which are in the group of AC inhibitors, casukinin, multifunctional, casophosphocaseins, immunopeptide, antibacterial. And our results correlate with the studies of also different groups studying these interesting peptides which contribute to human health and are very part of the diet of mankind. In conclusion, I would like again to say that bioactive peptides from food protein are very important for the mankind. They were studied many years. Many scientists take part in determining the detailed structure of bioactive peptides. They are derived from a wide range of foodstuffs like eggs, fish, meat, cereals. Also, milk and other dairy products are among the best precursors of bioactive peptides and have been mostly studied. The contemporary world is coming to terms with the ancient quote of Hippocrates, "Let food be thy medicine and medicine be thy food," than ever before. Thank you for your attention. If my lecturing was useful for you, I will be very glad. And here is small pictures you can see from different parts of Bulgaria. Bulgaria is a small country. To the right you see where we are placed in Europe. And also on in the down of the pictures, we can find very important Asian things, especially in the middles. This is the cave Magura, which is very important, and there we find pictures on the wall of the cave. And maybe at that time also people eat milk products and help to develop our knowledge about these wonderful products in the world.
Thank you very much, Professor Ivanova, for your presentation. Unfortunately, this was a record video, so she can see only after that. If you have a question, we can provide the email, so you can email her and ask her your questions. So we're a little bit ahead of our schedule, so we can have a little bit longer break for the people here to have the coffee and cake provided by Faculty of Pharmaceutical Sciences. And the next presentation will be at 11 o'clock. Well, she's already online. Maybe we can start a little bit earlier so we can have a longer lunch break. Okay, let's do the next one. Next presenter, it's Ivy. Do you want a small break? Okay, let's have the 5 minutes break and we come back with Ivy. Hello, Ivy. Can you hear me? So, Ivy, let's give for people 5 minutes break because it's not only you that like the food. People like food here, so we can have a cake here and a coffee, and then we can have you in a 5 minutes starting with your presentation. So see you in a 5 minutes. Hello, Ivy. Are you online? We cannot hear you. Not again. You're not allowed to. You're now. Try it now. Say something. No. Technicians. No, it's not working. We cannot hear her. Il microfono è dentro. Quando ela começa vídeo, sai?
Não, não vídeo. Qualquer vídeo tá rodando aqui no áudio.
Ivy, we think that your microphone is not something wrong there. No, we can see you try to say, but there is no audio for us. Okay, maybe it's a good idea. Eu consegui 22 artigos dentro do mestrado. Sim. Só que um ponto. Lá na Filipinas tem. Eles ficam muito vitro. Let's see if we can hear you then. No, it's not. So we was thinking that we can start early, but we can start maybe on time. Using the Macintosh. Hmm? Still not. We cannot hear you. Can you change the computer, maybe? Still cannot hear you. So please, you can use your cell phone and you can talk via the WhatsApp. So I can put. So. Okay, okay. Ivy, you need to talk on your cell phone, and I will put it via the cell phone, your voice here.
Is it alright?
Well, we don't have option.
Okay, I'll try my best then.
Okay, let's try in this way. Working?
I'm not on mute.
Ivy. No, we can. No, we can. So we can put it here in the isolated room. There is no, cannot be microphone here. So try to say something and see if they can listen in there. Talk something.
I'm saying something. Okay, yeah. I think you can hear me. I'm using two different computers, and I don't think it's the computer that has an issue. Because on the computer,
If she can use the Zoom to speak the presentation, you can capture the Zoom.
or
Ivy? Ivy Fugaban,
Still
can you
no
hear me?
sound.
Now it's work. Hallelujah. Say something. Say
Still
again
no
something.
sound.
Yeah, it's work.
Oh, it worked.
Okay, I will present you very quickly, so I don't want to lose your voice again. Next presenter is Joanna Ivy Irorita Fugaban, or just Ivy. She's from Philippines, studying at the university there, then works for short time there. Then she moved to Korea to have her master study there. One of the more productive students in Handong Global University. And then she ran away from Korea and stopped only in Denmark. And currently she's almost in the end of her PhD project. And it's a really pleasure to see you and to be with us today, even with some technical problems. So please start your presentation before we lose you again.
I hope not. I hope you're hearing me all right. Okay. So before everything else, let me go to my presentation, of course. And I hope you don't lose me anymore on any more technical stuff here. So good afternoon, everyone. So my name is Ivy, and today I will be talking about the multifaceted functionality of Bacteriocins from food to medical applications. So before everything else, with that very short introduction that we have, and I hope you get to bear with me along the presentation, with all the technical difficulties that we had, let's start slow and beginning. So my name is Ivy, and I am currently a PhD fellow in international flavors and fragrances. So I am an industrial fellow, and along with that, I am also affiliated with the National Food Institute here in Denmark, in Technical University of Denmark, to be more specific. And by profession, I am microbiologist. And my hobbies, aside from being a scientist, is that I do photography, music, outdoors, and I really love dogs. So I hope that inspires you to do something else aside from science. It's also nice to do that once in a while. So going with this, I hope you can still see my screen and everything. Is it all right? Is it doing all right? Yeah. So let me begin. More introduction, I guess. So my current project, and aside from the topic today, my topic today is my previous research interest. And my current research interest is focused on functional peptides, especially on plant-based fermentations. And this will be a bit of an overlap later on my presentation. So what I'm currently doing, just to give you a background on how diverse my research is all about, is we're focusing on plant proteins now and developing starter cultures and adjunct cultures for alternative food sources fermentation. So without further ado, let me proceed with my presentation. So this is the outline, and this has been shortened version of what I've been exposed and what I've been doing on Bacteriocins studies for the previous years. So today we will be talking about a short introduction on the Bacteriocins, followed by the basic background on nomenclature, classes, and mode of action, along with the isolation and methods of characterization or study of Bacteriocins, Bacteriocins applied in food systems, Bacteriocins in medical field, and the future applications, and the current gaps that we need to address or we want to further explore in the Bacteriocin research. So on the background, just to give you an overview of, or why are we actually interested in studying Bacteriocins. So Bacteriocins in general are antimicrobial peptides. And with this emerging need for alternative antimicrobials, especially on addressing the problem with antimicrobial resistant pathogens, it's quite alarming that this review written by Thakur and colleague have predicted that around 10 million deaths will be associated to antimicrobial resistance infection. And we all know that lactic acid bacteria, which has been discussed all throughout the week, I would assume, and also Bacteriocins has been well discussed in some of the lectures previously on this summer course. Lactic acid bacteria has been acknowledged or granted the status GRAS, which is defined or known as generally recognized as safe organism. This would allow them to be employed on various food systems, allowed to be ingested by human beings and animals. And aside from that, this microorganisms actually produces a lot of beneficial metabolites. Aside from the metabolites that they produce, they also functions as probiotics or starter cultures on various food products that we consume today, which are associated on beneficial functions or beneficial effects on the consumer itself. So we see from this figure that there are various benefits in drinking or eating probiotics. It includes decrease in cholesterol, which has been previously discussed by Professor Ivanova, and also immune modulation, along with microbiome or microbiota modulation, which is also a big research interest nowadays. So lactic acid bacteria produces a very functional metabolite called bacteriocin. And bacteriocins has been employed not just on the food preservation and antibacterial functionality, but is also further explored as anti-cancer compound and also used for drug delivery and among other infections. However, if we look on this graph, the study of bacteriocins has gone a long way. And bacteriocins, if we're going to be very particular about its definition, it is an antimicrobial peptide, which is typically effective against phylogenetically related microorganisms. And this should be highlighted because sometimes this definition or this particular nomenclature definition of bacteriocins has been overlooked. And if you see on this graph, we have quite a lot of studies that emerged on 1970s forward and a slow development previously. And with all the studies, we see still a very slow implementation and industrialization of bacteriocin application. So bacteriocins, which are typically named after where they are isolated from or which organism produces it, is typically based on how they are produced and how the structure is being observed after it is produced outside the cell. So that's the major classification basis for the types of bacteriocins. And to give you a brief overview of the classes, and I wouldn't expound so much on this because I assume it has been well tackled on the previous introduction. Classes of bacteriocins include variances across people who's writing reviews, who's trying to make new classifications, and such and such. However, on this presentation, I will be only tackling 3 classes of groups or classes of bacteriocins, including class 1, which is basically characterized as bacteriocins that is comprised of structures undergone extensive post-translational modification. So this modification is aided when they are actually produced by the microorganisms, and before they are actually exported out of the cell, they undergo various enzyme interaction that causes them to form some cyclic structures. And this is mainly characterized or observed on majority of the class 1 bacteriocins. With this formation of peptide-to-peptide bond that causes cyclic structures, this also leads to much more stable structure of this bacteriocins, thus allow them to be applied on a lot of different situations, applications, and industrial relevance that could be further explored on this group of bacteriocins rather. On the class 2, on the other hand, it's the opposite of the class 1. These do not undergo much post-translational modification. However, they are known to be thermostable. This is because they are usually small or short peptides, typically between 2 to 10 kilodaltons, and this is actually comprised of some of the enterocins and the pediocin structures. And lastly, on the class 3, these are usually the large bacteriocins, which is greater than 10 kDa or 10 kilodaltons, and usually heat sensitive. Some of the representatives of this group or class is actually associated with some carbohydrate moiety that would actually causes them to be unstable. So that is the main difference across the groupings of bacteriocins. So for the mode of action, I only have this image, and if you want it, you just have to let me know. I can also forward the paper where I got this one. And it actually summarizes very well the mode of action of bacteriocins. So if you look on the upper part of this figure, if you look here, we have various mode of actions, and majority of them are actually associated on the surface of the target microorganism or the membrane. So some of bacteriocins actually attaches to the lipid tube, which then interferes with the cell membrane synthesis of the target organism. Some of the bacteriocins acts as like pore formers on the surface of the target organisms, and once these pores are actually embedded and found on the target organism, instability on the target organism occurs, thus causing cell death. And some bacteriocins, like pediocin A, actually acts on the MAN-PTS or the transport system found on the surface proteins of this target organism. And it actually causes inhibition of the functionality of these transport systems, thus inhibiting some of the key metabolic activity of the target cell, hence causing death. So there's a lot of variances in the mode of action of some bacteriocins. Some of the less well-known bacteriocin we can see from here actually target the outer membrane, and some actually target periplasmic activity, where some cells doesn't have the membrane itself. That's a lot of complications and discussions on the mode of action of some bacteriocins. However, this is the general overview, and if you want a much more detailed one, I'm willing to talk about that. So proceeding with sources and known antimicrobials. So aside from bacteriocins, or aside from this small peptides that we have been talking for a while now, there's also a lot of antimicrobial compounds produced by lactic acid bacteria. And it's not also just lactic acid bacteria or the known classes of lactic acid bacteria that produces it, but also some representatives of Bacillus species are actually quite interesting in this field, particularly on the food application. So for some of the lactic acid bacteria, majority of lactic acid bacteria actually produces these metabolites, and some of which includes organic acid, acetylene, and diacetyl, which is quite known for flavor development and flavor association, phenethylamine, which is very associated to the Enterococcus group, and it's also known as a biogenic amine, and is now found out to have a very particular role in the gut microbiome signal peptide interaction. And some of the antimicrobial compounds also include hydrogen peroxide and carbon dioxide, which is produced during fermentation. And all these antimicrobial compounds actually contribute on the biopreservation or various applications such as antifungal functions, antiviral application, and among others. However, I have to highlight here that bacteriocins are definitely different from bacteriocin-like inhibitory substances. One of their main difference is that bleas have a broader range of target microorganisms compared to bacteriocins, which only target a very specific set of organism. So that's the difference. And sometimes bleas loses its activity when exposed in high concentration of peptidases or proteinases. So those things we have to actually be careful when you're trying to claim if you're working with a bacteriocin or a bacteriocin-like inhibitory compounds. So the current methodologies for discoveries of bacteriocins and studying bacteriocins in general have expanded and expounded for the past years. And if you're interested in accessing one of the pipelines developed in our group, we have this online. One of my colleagues has developed this very functional genome search for bacteriocin clusters, and it's available online for free. On this GitHub link, you can just have access on it and see and play around if you can do that. And you can do genome mining to search and discover some new gene clusters for bacteriocin expression. However, on this paradigm, if you see on the right-hand side of the slide, it's divided into three different groups, I would say. So the first one on the left, which is this one, this is the classical way of actually characterizing bacteriocins. This is the one that we do in the lab, wherein we isolate strain and then test it against microorganisms and then see which one actually works or which one doesn't work. And then if you see an inhibition zone against your target sensitive organism, you further characterize it in the lab and see if the structure, how stable it is, what possible application you can do with the bacteriocin. And that's the classical way of handling this bacteriocin discovery and characterization. However, with the emerging technology, now we have various ways to do in silico search for bacteriocin producers. And as I have mentioned, we have this pipeline developed. And what you usually do is that you get the genome sequences of no matter how huge data set you want to handle, put it on the pipeline, search for whichever have expressed the complete set of bacteriocin gene cluster that would enable it to be possibly encoded and expressed by the organism, and then do the reverse and go to the laboratory and explore it. And that actually reduced a lot of work on the screening on some of the works that's currently been done in this university or institute I am at the moment. And furthermore, with the information that you get in silico, you can also do a lot of engineering and functionality work with this bacteriocins. You can modify structures, change one peptide at a time, see how much it affects actually the target organism, how broad or how narrow it will end up, how stable, unstable it will be. Those are some of the studies being conducted in University of Cork, College Cork in Ireland. It's one of the discussions we had with some colleagues there who has been working with it. So yeah, the current methodologies in bacteriocin studies has expanded and has been it's continuously improving at the moment, and it's quite interesting field still for a lot of young scientists like us. So with that, now what? Like, why are we actually studying bacteriocins? What can we do with that? So there's a lot of applications wherein bacteriocins are isolated from food and also applied as food preservative. And as a start, like the producers are actually wanted to be starter cultures or adjunct cultures. So some of these are actually lactic acid bacteria, and majority of them actually are lactic acid bacteria, and they target Listeria and Staphylococcus. And observed to have a significant reduction. And this information has been taken from a review we have written 2 years ago. We have also identified that we want to have a strain that is a good starter culture and also produces a antimicrobial compound that would inhibit the possible growth of spoilage organisms. However, if your starter culture cannot do some of the functionalities you want, you can add adjunct cultures, which compensates the missing functionality that you have that you want your starter cultures to have. So this can be employed in various food systems, and the food industry is a really, really wide industry for this matter. Furthermore, the industry doesn't apply pure bacteriocins because that's expensive as hell, and you just wanted to have a semi-purified version of your bacteriocin for it to be efficient. So some of the commercially available bacteriocins at the moment are niacin A, Z, and pidgecin PA1. And these are the only ones I can find in the market at the moment. So as I have mentioned, niacin A and Z is currently marketed by DuPont Danisco, which is now IFF, where I am working at. So this bacteriocin or this product can be bought online if you just want to try it out. It's there and it's available. And if you want to do like homemade fermentation and stuff, this is also nice. Furthermore, there's another version of niacin AZ combination, which is called Chrysine, which is also produced by a Danish company called Christian Hansen, which is now known as Nuvonesis. And I think they also have a company there in Brazil, Nuvonesis. And this particular bacteriocins are actually claimed to be plant-produced. So that's a very interesting claim on this particular product. And I've tried to read a lot about it. However, of course, with the trade secrets and everything, the production itself is not disclosed. However, the next bacteriocin, we have pidgecin PA1. And this is commercialized by Kerry Group. And the product is called PA Alta 2351. I searched for the second version of this. However, this is the only commercially available product I can find of pidgecin now. So with all the researches done in bacteriocins, we're still logging on the actual real life application of this very powerful peptides out there. And I hope that in the future, we can see much more of this in the market and being used by normal people around us. And that's why we do science. It's because we don't want it to end just on the paper. We want people to use the technologies we're developing. So the further application of bacteriocins, as I have mentioned, it's because they have a very narrow spectrum. They are very good for targeted treatments. And one of which is actually their ability to help facilitate a niche establishment on IBS patients. So if you know, there's a huge ruckus now on the research in microbiome, and it's always difficult to have a proper solution for the existing problems with that. And this is one of the possible applications of bacteriocins on this field. Furthermore, there's a lot of also researches associated to their clinical applications, including their effect as spermicidal and possibly contraceptive product. Excuse me. Their ability to inhibit oral health pathogens, their association with cell toxicity, which can be further explored for anti-tumor, anti-cancer agent, and among others. So to give you a concrete example on this clinical application, we conducted a study in South Korea when I was doing my master's on some of vancomycin-resistant Enterococcus, which are clinically isolated. And we wanted to see if the Bacteriocins I have been studying, or we have been studying, are actually able to inhibit this biofilm formation of clinically isolated VRE. And if you see from here on the right-hand side, this is actually the viability assay. And a certain concentration totally inhibits viable cells within the biofilm. And that's actually very impressive, as biofilms acts as protective layer for this highly potent microorganisms. So we wanted to see further or confirm this observation via flow cytometry, and we have measured corresponding viable cells or viable cell count on this observation. So this is just an overview of it. Another study conducted also in South Korea by Kim and colleague, which is one of our master students when I was there also, did a challenge study on oral pathogens. And we have observed a significant reduction or a nice result on the challenge versus Streptococcus simulans, which is a known oral pathogen, by various lactic acid bacteria, including Streptococcus salivarius and also a Lactobacillus plantarum and Lactobacillus gasseri strain. So here you can see a first-hand possible applications of Bacteriocins. So now what? Like with all this, okay, there's people who's doing this research, doing those research. Now what else we can do? There's still a lot of things that has been left unexplored. And especially now that we have a current race to find alternative food sources with the increasing demand for our plant-based foods, alternative meat sources, alternative protein sources. Those things actually have directed us to find an alternative inhibitory compound for the spoilage organisms that is associated to them. So with this new food products comes new spoilage organisms. And as observed by some colleagues, these spoilage agents are not the usual or typical spoilage agents that we observe from the typical foods that we have at the moment. So the need for starter cultures that is also able to inhibit these microorganisms or spoilage organisms is important. So we need a lot of starter cultures that is optimized or adapted to plant-based foods that would be at par for organoleptic properties, texture, flavor with the current products that we have in the market coming from the animal food sources, and some anti-nutrient factors which are actually much more concerned or much more concentrated problem on the plant-based or alternative food problems. So this is something that we have to take a look at in the next years to come. And this has been one of the major focus of researchers now in Europe in general. So to give you a background on how difficult it is or how different it is between the fermentation in the plant basis and in this actually previous or traditional food sources that we have. So for example, if you take a look here, we have a cheese ripening process. So in a milk, you have fat, sugars, and proteins, which also been discussed by Professor Ivanova on her lecture. And we see here that the functionality of lactic acid bacteria usually occurs in lipolysis, a way of degrading these fats found in the milk, glycolysis, which actually converts the available sugar on the substrate to a functional product at the end of fermentation. And the most important part and the most difficult part in the dressing is protein degradation. So proteolytic strains are actually very difficult to find, especially for plant-based. The proteins we found on animal food sources versus on the plant food sources are different. and the way this organism actually catabolizes or change the structural conformation or convert one product to another is different. And we want them to produce the nice compounds that actually helps in development of flavor and nice texture to be present, which is very difficult to find. So the traditional starter cultures and adjunct cultures that we have been using for the longest time is now not effective on these substrates. And some of the major enzymes that we have to take a look at, and if you're trying to isolate a functional strain, these are the things, aside from the bacteriocin production, these are also some of the things that you want to look at when you're trying to search or discover new starter cultures in the lab. So some of which actually is the production of enzyme called aminotransferases. And this is the most important enzyme on cheese production because it actually gives the cheese flavor, which is the glutamate. The decarboxylases, which actually leads down to the production of aldehydes and carboxylic acids, which is important on a lot of fermented food. And also this alcohol dehydrogenase compound, which is important in beer fermentation, wine fermentation, and among others. And also the keto dehydrogenase, which also helps in production of some flavorings. So furthermore, aside from the food production, what interests us now in the medical application, as I have mentioned on some previous slides ago, bacteriocins are good in facilitating niche establishment in the gut microbiome. However, the problem is the successful delivery of these bacteriocins to the gut is still a problem. And in order to address that, we need to find a functional and non-invasive way of doing it. So that's still a lot of questions that we need to do. And furthermore, understanding much the mechanism of action of these bacteriocins as immunomodulatory compound is also still a question. So with all these gaps that we identified, how can we actually leverage our current knowledge and the knowledge that we don't know on our current research? So understanding the mechanisms of bacteriocins and how they react and the arising problems, actually, particularly resistance, as antimicrobial resistance, like antibiotic resistance, is arising, the same is true with bacteriocins. And continuous development, structural development that is acceptable for the market is important. And one of which is actually adaptive laboratory evolution. It's a way of naturally evolving a strain until it produces a certain compound or structure of bacteriocin that would still kill the resistant bacteria from the previous version. So that's a way of actually leveraging or finding or addressing these problems. Furthermore, we still have a problem with the starter cultures and adjunct cultures applied on particular sets of food. So as I have mentioned, we are now shifting from the traditional food sources to a much more sustainable food production and food matrices. And this would actually allow us to discover much more of what is working and not working on these two different facets of food production. And furthermore, the action towards proving the efficiency and applicability of bacteriocins in the medical field should be further pushed through. Because if we could see it, there's still a stagnant improvement on the application of these bacteriocins as an alternative for antibiotics per se. And this is quite sad in a way because there's a lot of research, good research out there that's been conducted on this. However, the governing bodies are still not looking into it and trying it out and pushing it to be employed and used on these fields. So as I've mentioned, we still have a lot of concerns and limitations with the use of bacteriocins. And I would like to reiterate that as we go and push through with the research and the study of bacteriocins, I hope we can also push the lines for the application and acceptance of this antimicrobial peptides in the market and in the medical field. So that's all. Thank you. And I hope you had a nice time listening to my lecture. So if you have any more questions, just let me know.
Sanida,
Tusintak.
thank you very much for your presentation. Well, now we have time for questions.
Yes.
Questions from the internet, from the live stream, we don't have it. But I'm here with some colleagues that work with the bacteriocin. Maybe there will be some questions from the audience. Okay.
Hi, Fugaban.
Yeah, hi.
Thanks for the presentation. And my question is for you both. Sometimes bacteriocins can use it without the intention of killing the bacteria, but reducing the virulence. Yes? In this case, is considered a kind of antivirulence strategy. And my second question is, do you think the combination of bacteriocins with antibiotics is a good alternative to reduce antibacterial resistance?
Yeah, for your first question, some of the bacteriocin mode of action is actually just bacteriostatic. And that actually is a good factor in a sense if you combine it with something that actually kills the organism. Because if you just keep on like treating it in that way, it doesn't actually help much in the infection or in the amount of pathogens in the environment. So yeah, it actually is affecting the antivirulence of the pathogen. However, it's not most likely what I want if I want to treat that infection. On the second question, however, can you please repeat the second question because I'm a bit...
Yes. Do you think the combination of antibiotics with bacteriocins is a good alternative to reduce antibacterial resistance?
Yes, thank you for your question. We actually had a study on that, quite a lot. And we observed that it is nice, but you have to choose the correct antibiotic to pair it with. Because you can pair bacteriocin with another antibiotic of different mode of actions to actually compensate them together for a better observation of functionality. Because as I observed on previous researches we conducted on this, if a resistant microorganism is actually being treated with a bacteriocin with the same mode of action as what the bacteria is resistant of, it actually doesn't work. So you have to have it as a combinatory in a smart way, at least, for it to function.
To find the right couple.
Yes.
Right bacteriocin with the right antibiotic.
Exactly.
Thank you.
There is more questions from the audience. Do we have questions from online? I think everyone was looking for the lunch because it's not only you attract to the food, but people here in Brazil as well. And it's like 11:45, everyone start thinking about the lunch.
Yeah, I love food and started making money out of it.
But it's not fair. You never get fat. It's like you can enjoy the food. What's the secret? To have the right bacteriocin in your body that can control your metabolism.
Yeah, it's just genes.
We have actually. Do you think that that is interesting to study the production of the bacteriocin using agro-industrial waste?
Well, it's always interesting to be innovative on where you actually isolate your bacteriocin producing organism. And the less invasive your sources are, or the more naturally occurring the sources are, the more diverse microorganisms you isolate. And the probability of you getting actually a good type of bacteriocin producer is quite high. And the way, yes, like waste management, I wouldn't say I'm quite particular on how you actually would do that. And I wouldn't say I'm qualified to assess the application of bacteriocin on waste management because we never did that. So I really don't know yet. But that's
But
an interesting application.
some
Like-
waste products from the food industry can be used as a source for the growing the lactic acid bacteria produced, because we know that the cheese whey can be used as a source for the growing the bacteria
Yes, for
produced.
the
We can use
isolation, yes.
molasses. Yeah.
But I wouldn't much leverage on that because there's much more better use for the wastes produced in the food industry rather than actually isolating this or using them for a very small part of research or discoveries.
The cheese
Yeah.
whey was in the past giving to the pork to grow the proteins as a food for porks. This day we're giving to the people in the gym. So
Yeah, well,
the option.
I mean, in industry wise perspective, if I'm gonna give that perspective, I wouldn't do that in a sense. Like I would explore it on a different way. So yeah, I would do something else with it to be
Well,
completely honest.
most of these waste products, actually byproducts from the food industry, they can have a new life because we can use
Yeah.
them as creating a new food products and extracting the right components from them. This is actually a trend in the food industry in these days.
Yes, and we are actually doing a lot of research on that. That's why I'm saying it's not the first thing that came on my mind because there's a lot of other ways that you can use it aside from that. Yeah.
Well, a good example was the cheese whey, first for the porks, now for the gym. Okay, but thank you very much, Ivy, for your participation. It was really nice to see you smile there. And
Yes.
in some Denmark, and I hope to see you soon in some of the meetings around the world, because it's
Yes.
really enjoying when having discussions with you. And just want to mention that she was winning several winner of the best presentation in several conference. So it's like competition for you if you present together with her. There's another question from, yes, we have another question from the internet. Sorry, we can ask you. Can you
Yes.
put it? Anna Clara Kuchik. The source of bacteriocin can limit its application. For example, a bacteriocin isolated from the gut-birth bacteria
No,
can be in food.
Well, that's of course a different because the other one is talking about food waste, and this one is entirely different perspective. Because if you're going to, it's not actually limit its application. It's much more of what you can do best on the current product or the current thing that you have at your hand. What are you gonna use it for better, right? But this actually got sources for bacteriocin producers. It's actually a good source per se, especially if you want to apply it particularly on the same environment, because organisms definitely isolated on the same systems would proliferate better when you apply it on the same system by principle. So yeah.
And just need to be safe, the bacteriocin, and we can apply it where we want. Right.
Yes.
Okay.
And yeah,
Tell me.
also just to add on this question, I have a colleague that did insect gut microbiome research, and he actually isolated quite a lot of bacteriocin producers on this environment. And yes, you can apply this bacteriocins in the food system. However, what we observe on this particular set of bacteriocin producers is that its mode of action and its actually spectrum of activity is very limited because of the environment. We concluded that it's because of the environment we found this producer from. So yeah, it can be applied with a lot of things, but we have to consider those.
So thank you very much, Ivy, for your presentation. It was a really pleasure to see you, to hear you talk. And we're going to have a lunch here, and you most probably are going to have your dinner in Denmark. So
Yes.
enjoy, and to see you in the next occasion soon. Thank you very much.
Thank you, and thank you for bearing with me on my technical issues.
Bye-bye. Bye-bye. Hello everyone. Welcome to the last session. A little bit more and we're going to be finished. Santosh, can you hear me?
Yes, Professor Todorov,
Okay, this is a good point because we had some technical problems with some of the other speakers. We had a video but not audio. So, okay, let's continue. The next presenter that we're going to have it today, it's working in a very specific area. He worked with the Bacteriocins, but he worked with a focus on the group of archaea. It's a very specific group of microorganisms, reasonably not too long time ago, put it in the literature. And as well, in the work, Professor Santosh Tiwari was showing that some of these antimicrobials that actually, some of these microorganisms of archaea, they could be actually a very good producer of antimicrobial compounds. And they have a very good potential related to the fact that they're coming from the very specific ecological niche. And this is a very specific group. So Professor Tiwari, it's from University of Maharshi Dayanand University in India. He has his experience as well from the Rutgers University in the US, where he was working with Professor Chikindas. He's extremely actively working in the antimicrobials, and it's really a pleasure to have him today with us because going to put another point of view on the antimicrobial peptides, especially this one from not very well-known microbial group as archaea. So thank you very much to be with us today. And now it's a time for you to present your talk. And don't worry, you can talk how long you want because the next presenter just claimed that he cannot present today. So you have even more time if you want. Thank you very much to be with us. And now it's you. Thank you.
Thank you very much, Professor Todorov. Am I audible? Please confirm. Is my voice audible?
But slides, we cannot see it yet. We just see you a screen with three black spots of the cameras, but not yet your slides.
You can't see my slide?
No, not yet. You need to just share the right screen when you're sharing, because I think you share the principal screen of your computer.
Is it visible?
Not yet. No. Now it's again the principal screen.
Okay, let me change.
First, put on the big screen the presentation and then share.
If I go to present and then share slides, I'm getting, you have no slides. Okay. I think if I share my screen, then it should go.
Just try it.
Okay, I'm sharing my whole screen and it should present my screen. Let me try. Also share system module. Can you see my slide?
Now it's working. Perfect. Excellent. You can start. Thank you.
Thank you very much, respected Professor Todorov and my colleagues. First, I would like to thank Professor Todorov for organizing this wonderful event, which is a topic related with the kind of a service to the humanity. And his contribution to organizing such kind of events is not only initial leads, but will have a breakthrough in the future direction to discover novel bacteria, beneficial bacteria, and to discover novel metabolites, which our society is facing. Several problems, not because of food, but because of the medicines also. So for you, it is a post-lunch time, and for me, it is a post-dinner time. So both the times, this duration is sleepy. And I'm in between a sandwich between you and me, because if you have a food and energy goes to digest the food and your mind becomes sleepy like that way. I will try my best to make you awake and connected with me by understanding some new things what we have. Right from the initial research, we are involved in the understanding of role of bacteriocins, of probiotic bacteria, their beneficial roles. Most of the works at this seminar or in this session also, you have seen the bacteriocins. The theme of this discussion was mostly, I was looking at this how these bacteriocins are related antimicrobial peptides can work for better health, for better food, for quality enhancement. My friends, whether you accept it or not, the world climate is changing. It's moving drastically from lower to higher degree temperature and different extreme conditions. The conditions are changing day by day, and because of because of the human interference, lots of human activity we have, and there is a climate change and increased temperature has been recorded. You won't trust that in India we had in this summer 50 degree plus, 50 degree centigrade. And if such temperatures are increasing, humans will also have to adopt. Either they will do or die. That kind of situation is going to come, and only those will survive who are having that kind of capability to resist or survive under the extreme conditions. So I thought that in addition to probiotic bacteriocins, I should work on those metabolites which are stable in a very extreme conditions, and they will provide some clues that what are the actually metabolic activity in the cell which are responsible for such kind of high resistance. So my group is entirely working on probiotic bacteria and the bacteriocin part. We have one system, one stream, which is working on the different level like archaea. Archaeas are entirely different than the bacteria, and their level of understanding is very poor as far the literature is concerned. We know a lot, but not as compared to bacteria. So in next few minutes, I'm going to tell you something about how this archaeal group, like haloarchaea, we are having examples of here. One is how haloarchaea behaves and what are the specific metabolites we are having. They are equally good like bacteriocins or other metabolites or probiotics which are being considered for a good research. Next, I'm changing my slide, Dr. Todorov. It's okay?
Perfect, it works.
Right. Hope you are looking at the, I'm changing slide, right? So this haloarchaea are microorganisms which are living in a very extreme conditions. We are having a very desert situation in India, and also throughout the world. You are having very desert salt lakes where such kind of halophilic microorganisms are growing. You are having halophilic bacteria and halophilic archaea, which domain is different than the plants and the bacteria and the animals. They are strictly anaerobes to facultative anaerobic bacteria, and the beautiful feature their survival is they are able to tolerate up to 30 degrees centigrade, 30% NaCl. I'm sorry, 30% NaCl is something which is a very saturated conditions. And more than 30%, if you go for that, it become a crystalline. So these microorganisms are able to resist very high salt concentration, and they are able to grow. And alkaline conditions are favorable, and also 30 to 50 degrees centigrade temperature they are able to tolerate, and they contain pigmentations to protect themselves from the UV radiations and other extreme conditions. These carotenoids, the pigments, which are very much helpful, and they are natural. And I mean, some of the initial research have shown good properties which are beneficial. So I will concentrate more on the antimicrobial peptides which are secreted by such kind of microorganisms, haloarchaea. It's similar like a Bacteriocins. What are these small peptides from haloarchaea? We say technically it is a Halocins, but it's like a Bacteriocins which are secreted from the bacteria, but were very least known. It's not mostly studied. But in our group also, we started a few years back thinking that some haloarchaea are also producer of such kind of antimicrobial peptides. These peptides are secreted in the environment whenever this organism grows into like bacteria, and they inhibit generally related strains. You know, it's a competition between the related strains. If two strains are growing in the same ecological niche, they will be inhibited, like it is shown here in the slide, that there is a competition from the similar group. It's not from the entirely different group. If you see the environmental perspective, it inhibits mostly the related strains. It's smaller in size, and mostly few of the halocins, H4, C8, and S8 are reported till date, but mostly they are unknown. And this area is something which can be explored. There are several application point of view which can be considered. Some of the workers, they have identified this area that yes, we are having a very salted food, and normal proteins, if you take from the bacterial system or plant proteins, sometime at a very high salt concentration they get precipitated. But such kind of halocins are very stable at, as we have seen, 30 to 20 to 30 degrees centigrade. It's 30% NaCl. Actually, they are able to tolerate. So that can be used as a food preservative. In several leather industries, it's a kind of contamination by the haloarchaea, and their hides are damaged. So such kind of use of halocins in the leather industry may help. There are few research which on the halocin S6, which is able to inhibit the sodium hydrogen antiporter in the ischemia conditions, and it's a great therapeutic agents where this peptide can be used. So there is a helpful, very less study on the structure function relationship of this peptide in the environmental conditions. So considering all these things, we thought of that we should screen some of the haloarchaea which were isolated from the salt lake we have in one of the state in India. In the western part of India is very dry and desert, and it has salty situations, and most of the lakes are very dry and crystalline at that condition. So my students, they collected these samples and screened them for antimicrobial activity, and found that out of many, we got few which are very good producers of antimicrobial compounds. You can see in the right side of this image, there's a very clear zone of inhibition recorded. Some of them were low producer, some of them were higher producer, and some are the nil, no activity. So we got a mixed kind of reaction from all those isolated stains, and that gives a situation to be as hopeful that yes, we are landing up to a situation where we can collect some of the antimicrobials producing haloarchaea. So we selected few of them to understand. And before looking at this, if you see the growth profile and halocin production, so if you see this green line is showing the graph growth, and red one is the halocin production. So mostly it's a growth associated phenomena. When the organism grows, it's like produces higher, but as soon as it proceeds to the stationary phase, the activity is declined. In some of the cases, some of these strains it's stable, but in others it's like a very low. So once the activity goes down in the exponential phase and in the stationary phase it decline, it is speculated that in the stationary phase, when cells dies, it releases some of the proteolytic enzymes, and these halocins are mostly proteins, and they are degraded in the stationary phase. So later on it is degraded. So one has to be very quick and responsive to collect the fraction from the R's, where is the maximum production is there. So based on this higher activity, my group selected some from this HA1 and HA3, which were showing the higher production level of halocins. So these selected isolates, if you can see the color, just I was having to show you this slide. It's a very nice pigments. It's like carotenoids, and these carotenoids are pink and red colors, which are also having a very good therapeutic property. I'll show you later on. But halocins, like we take a cell-free supernatant and put it on the target strain, and shows a very clear zone of inhibition against the target strains. We are using haloarchaea as a target for this as an indicator strain, and that shows a very good clear zone of inhibition, and that gave us some hope to us that yes, out of these strains we are having something which are producing high level of activity units as compared to other strains. So we selected these and try to characterize them, to identify at the biochemical level and molecular level. I have just shown you the comparative of these two strains. One is HA3 and another is HA1. If you see, this is the colony morphology of HA1 and HA3. HA3 seems to be more pigmented and reddish in color, whereas HA1 is something lighter, yellowish. If you grow in the flask, in the broth condition, their pigments is very clearly visible. This is the microscopic examination of the cells. Right side, cellular morphology have seen, and they are pleomorphic in nature. They are something which is, the shape is not clear. It's sometimes a triangle, sometimes basilar, sometimes roundish. So it's reported in the literature also that such kind of haloarchaea are having a pleomorphic shape. So because of this pigment, we are interested to purify this pigment to a homogeneity. As Professor Todorov also mentioned in one of his presentation, that you need a purified metabolites, purified compounds, if you think for the application. So in this case, it confirms that when we run this, we've separated these carotenoids, and this was confirmed by the thin layer chromatography techniques that one of the peak is completely, it belongs to bacteriorubrin, which is a well-known carotenoid reported by several haloarchaea. This bacteriorubrin was purified to a single band here, and it shows antimicrobial activity in this. So the carotenoid which is produced by this has a very good applications also. It's just like a kind of natural food additives as compared to the chemical additive whatever we are having. This will have advantage over it. So second feature, what we try to understand the concentration of different salts, how these organisms are growing, H1 and H3, and you'll find that it's very easily tolerate 15% NaCl, but it is able to grow at 20%, 25%, and 30%, almost like more than 40% growth is stable at 30% NaCl. But maximum growth is, the optimum condition is 15%. In case of H3 also, so mostly it's easily able to tolerate this 15 to 20% NaCl in the medium. And MgSO4 also, if you go for 0.3, so almost it's able to tolerate this level 1.8 mg%. And if you see the pH, mostly it is stable in the range of slight acidic to slight alkaline, and 7 or slightly alkaline condition is most favorable. 7.2 we have seen it's most optimum in case of this, and also in case of H3 this condition is 7-8. So it's happily growing between pH 7.2 to 8. If you see the temperature, its optimum is 42 degrees centigrade, but it's able to grow at 50 degree also, almost 80% growth at the 50 degrees centigrade. But in case of H3, it's almost 90%. This is growing equally, almost equally at 42 and 50 degrees centigrade. So it's thermotolerant and NaCl tolerating halophilic archaea. We took some of the tests to confirm whether these are archaea or bacteria. So in the Burgess manual, we compare that there are some carbohydrate fermentation profile of this organism is completely matching with the Halophyrex genus of the haloarchaea. So this fermentation profile we performed and compared with this group and found that they belong to haloarchaea. At the same time, most of the haloarchaea are catalase positive. In this case also we tested the catalase test and found that these are catalase positive, which is a unique feature of archaea, whereas if you see the probiotic lactic acid bacteria, they are catalase negative. So it's in contrast to that this archaea level is catalase positive. They are able to hydrolyze casein and starch, which is also a unique feature of this strain, and they are not able to utilize arginines. And there's no vicin sensitive property. These are the characteristic feature of haloarchaea we have seen and compared that the strain whatever we have isolated belongs to group haloarchaea. We move to little bit at the molecular level to understand whether we are having a bacteria archaea, because that was a main concern of the researcher, that probably we might have a halobacteria because they are also able to grow at high temperature and a high salt concentration. But if you run the methanolysates of the whole cell organism, if you take the whole cell methanolysate and run on the TLC, it's like a GDIM glycerol diether moiety is the unique feature of the archaea only, that the G-dem moiety is only present in the archaea and not the bacteria. If you see, we have these two conditions, to 1 and 2 is the archaea sample, and another we have taken 3 to 7 is the bacteria. If you see, this G-dem is completely absent in the bacteria, but it is present in the sample what we had, haloarchaea, HA1 and HA3. This is completely well studied, that the G-dem moiety is present in the archaea system, whereas in the bacteria we are having FAME, fatty acid methyl ester. So in the bacteria we have ester, which is absent in the archaea, whereas in the archaea we have diether, which is absent in bacteria. It's a contrast. So this gives an confidence to us that yes, we are on the right track, and probably the stain what we have isolated from the salt lake belongs to the archaea, not the bacteria. When we run this band G-dem to the FTIR, and FTIR gives a spectrum near which is ranging to diether, and which that confirms the presence of G-dem moiety in the HA1 and HA3 archaeal strains. We move to this step ahead, which is a very general test people use for 16S RDNA sequence analysis to identify archaea and bacteria. We use the same like PCR primers, which are specific for the archaeal system, 16S RDNA sequencing. And after the sequence, when we analyzed this, and after the analysis, we found that this HA1 belongs to Halophyrex larsenii JJ206, and HA3 also belongs to the same Halophyrex larsenii genus. So the complete 1500 base pair sequence were amplified, and after sequencing, the blast analysis indicated that whatever the sequence is, it's almost 98% is exactly similar with the Halophyrex larsenii, which confirms that the strain HE1 and HE3 both belongs to haloarchaea group. So it's identified that it is a Halophyrex larsenii. We have submitted this one of the strain HA3 to the culture collection center. They follow their own pattern to identify it, and they confirm that yes, the Halophyrex larsenii is the genus which have isolated from this. So the next story comes to after the identification that yes, what are the things we are having in mind. So as you know, I was mostly researcher of the Bacteriocin. I first touch from my side was I thought that I should go for the cell-free supernatant and test whether any antimicrobial peptide is present in these or not. So we followed this one by one, took the culture filtrate or cell-free supernatant, then we followed the ultrafiltration. We use separate these metabolites from 3 kilodalton to 5 kilodalton, 10 kilodalton, a specific cutoff size membrane you can use, and then we use gel filtration like a chromatography, gel filtration, ion exchange chromatography, and then finally the HPLC to confirm whether it is important or not. So whether it is a Bacteriocins or Halocin, as we understand this point, that it is very important to have a purified compound in hand, so that not only for the application point of view, but also for the characterization, understanding the property of this molecule, we need a purified fraction. So we asked to the funding agency, they provided this machine like ultrafiltration, where we use different cutoff site membranes. One can use 1 kilodalton, 3 kilodalton, 5 kilodalton, 10 kilodalton, so on, depending on the case. Like in one of the case here, we have used 10 kilodalton membrane filter. So if you pass with this 10 kilodalton filter, one is something which is opposite called retentate, and then which goes outside of the membrane, which is called permeate. And if you see, we have run this, the retentate and the permeate both and the retentate is showing antimicrobial activity, but the permeate is not. It means that the protein or peptide, whatever it is more than 10 kilodalton size, which is retained by the membrane. And in the permeate, you are losing most of the salts and other things has gone after ultrafiltration. So you are able to remove most of the metabolites from there. So it's a kind of partial purification. Once we had this fraction was after ultrafiltration, this fraction was run on gel filtration chromatography, and you see this is just a self-release supernatant, and see the several peaks. I mean, it is surprised that the cell is producing so many proteins outside. If you see these earlier fractions, there are hundreds of peptides. They are present there, but no activity. At top side, we have seen the activity. There is no activity in these earlier fractions, but the activity was in the later fractions. So, I mean, whatever we had, this the antimicrobial activity was present in one of the fraction, which is more than 14 kilodalton protein. That may be a protein which is antimicrobial in nature and which is a secretory by the organism. This is the basic definition of bacteriocins or halocins, that it is the secreted by the organism, smaller in size and proteinaceous in nature. So these fractions probably they belong to halocins, because it confirms that these are the proteins and they show the antimicrobial activity. But if you see the earlier fractions, so such kind of microorganisms, they are probably secreting not only one peptide or one protein, but so many. They're probably not showing antimicrobial activity, but their function may be different, which is a completely unexplored. I mean, just that kind of opportunity we have if we go to extreme environments. We'll get a novel microorganisms and novel metabolites. And this is how, we discovered tech polymerase. It's a kind of a boom to the biotech industry after coming tech polymer. A lot of people have amplified so many PCR is still being used. So that is also a kind of extreme level stability molecule. So we thought that whatever we are having in the HA1, almost similar profile was observed in case of HA3. You are having so many proteins which are present in the earlier fractions, but slowly we resolved this and got a single band, which was in between 14 and 20 kilodalton around, and this size protein was resolved using a gel filtration chromatography. And we thought it's mostly it is reported anionic. So it's a kind of comparison. Like bacteriocins, we are having cationic, but in case of halocins, they are completely anionic, and they bind to anion exchanges. Whereas mostly in bacteriocins, we use cation exchange chromatography. It's a strong cationic peptide, but this doesn't bind. It binds with the D-Chef route, like an anion exchanger, which we followed. And on the anion exchanger, this halocin, you can see it's completely binds and got a single band separated. And this was confirmed by a bioassay, which is showing a zone of inhibition. The molecular weight was found a little bit higher than the 14 kilodalton, as observed on the STS page. So this pictures probably landing you to confirm that whatever we are having from the secretory peptides, from the HA1 and HA3, this is something which belongs to approximately 14 kilodalton proteins, which is purified using gel filtration and ion exchange chromatography. And ion exchange chromatography to similar to this, which is confirmed by assay, that the peptide which is available is showing the zone of inhibition. And we thought that we should understand whether it is completely purified it or not. And that fraction was run on the HPLC and found the single peak. So it's just kind of confirmation that whatever we are running from the HA1 is showing a single band. and its bioassay is kind of antimicrobial activities. It inhibits the bacterium indicator stain. Put it here in case of HA3 and HA4. Both were almost 14 kilodalton peptides, and they are showing antimicrobial activity against the indicator stain. So this is where we are sort of confirming that yes, this peptide probably may belong to the halocins. The purification was compared by different technique. What we are having, the filtration, gel filtration, ultrafiltration, gel filtration, and action chromatography. So the yield is very poor, as in case of Bacteriocins we are having, and it's like a 10 to 12% of the total protein you can go for having. If you are doing chromatography, you have to lose most of the proteins, but need a proper optimization. If you optimize the condition, that can be done at the industrial scales also. But initially we did, and this much protein was sufficient to characterize the halocin what we are having in the laboratory. And we thought that we should check first the stability of this molecule, and this whether it is belonging to halocins or not. We perform certain stability assay of these halocins which we are having, and found that if you see the temperature stability, it's like up to 80 degrees centigrade in HA1, but 100 and 120 degrees centigrade, it's almost 50% activity retains in case of HA1. But in HA3, it's generally mostly stable at 80 degrees centigrade, but at 100 and 120 it goes down. In pH stability, it's a broad range. It goes from 4 to 12 degree. It's 12 pH. From 4 to pH 12, it's almost stable, so it's a broad range. And we have tested in most of the organic solvents, the halocin was able to be stable, but in some of the cases like ethyl acetate and acetone, the activity goes down. But in most of the organic solvent which was used, this peptide was able to tolerate. Further, because of the proteinaceous nature, when we use this trypsin and proteinase K, the activity drastically goes down because it's degraded, but it's resistant to the pepsin. So this confirms that whatever you have is a kind of proteinaceous in nature and stable to high temperature. This is the stability in the NaCl condition we have tested, that from 0%, 5%, 10%, and 50%. And this suggests that the minimum you need 10% NaCl to demonstrate the activity or stability of this molecule. But if you decrease the level of NaCl below 10%, like this is in case of 5% and this is in case of 0, without NaCl. If you see without NaCl, after 24 hours, it's more than 50% activity goes down, and it's completely denatured without NaCl. If you put some NaCl, its activity is maintained. Like at 10%, it's almost stable to 15%. So this molecule needs salt to make it stable. In both the case, like HA1 and HA3, we have seen more than 10% NaCl is essential for maintaining its stability. So it's a kind of evolutionary process. The molecule and the organisms is able to tolerate this kind of NaCl, this percentage, by modifying the structure. This slides represents the inhibition assay, where if this is the control, as compared to the control, if you treat with the MIC level and the double MIC level, its viability goes down. And at the same time, these fractions were tested on the scanning electron microscope, and this is the control cells very nicely if you see the entire boundary. But after the treatment, if you can see the membranes are ruptured, and if you use even higher concentration, it's a completely burst. At double MIC, if you see the So it's a kind of concentration-based inhibition, and similar to Bacteriocins, it's like it's a membrane-acting peptides. In this case also, the indicator stain is targets membrane. Some we found similar observation in case of HA3 also, that you are having a cell. We are using similar related stain of a haloarchaea HA10 as an indicator, as a target. So we have seen the activity against this stain, and found if you see increase the MIC level, its cells are something having disturbed morphological appearance. They are constricted, and mostly the dissipation takes place. But if you see the double the MIC level concentration, most of the cells are completely lysed. And if you see the zoom condition, we have seen some dissipation takes place and complete constriction of the cells takes place. So it's we are able to confirm that this one the molecule we're having is antimicrobial in nature, proteinaceous in nature, and probably it belongs to group Halocins. Yes. In this, you can see we have taken a host range, a few targets, and found that bacteria are not inhibited. So, I mean, as we admit this, it inhibits related stains. This is the reason that these haloarchaea are getting inhibited by HA1, and none of the bacteria were inhibited. So it's probably might have application with that range, that you can eliminate archaea, haloarchaea from any system, then you can use this specific peptide in this way. But HA3 is not able to inhibit even the haloarchaea. Only in one case HA10 was inhibited, but others were not, neither bacteria nor haloarchaea. So it's a kind of, we are having something like a very narrow range to middle range to the broad range. So one has to select for its application that what type of antimicrobial peptides or what type of halocins you need for what kind of application. It's a kind of personalized. So we thought to understand this, the peptide, we purified this halocin and did some multi-top-MS analysis. And after the analysis, you can see there is no database which matches 100% with the sample what we are having, HA1 and HA3. It's a kind of only 20% sequence coverage reported with the halocyanin-like precursor protein from the Halophilus larsenii, but no homology was observed with any halocins which are reported in the database. So probably it indicates that the molecule what we are having in hand is not matching with the database and maybe completely a new molecule. In the beginning, it shows like you are having a N-terminal sequence first, and this N-terminal sequence, when we did kind of blast analysis, there was no homology with the known halocin, whatever is reported in the database. So these even N-terminals were entirely different. Most of the time, the Bacteriocins N-terminal is the conserved, but in this case, this N-terminal is not matching with the existing database. So we thought of this as something. The halocins are from this strain, HA1 and HA3. These strains probably producing a unique halocins which are not reported in the database. Whatever these halocins are reported in the database, we try to design the primers, and these primers were used to amplify the HA1 and HA3. We amplify these two bands of HA1 and HA3 using this specific primer, and this kind of the sequence what we got from the HA1 and HA3 are having lost and analyzed this phylogenetic analysis and found that this HA1 was not matching, but HA3 is matching with the kind of hypothetical proteins of Halopherex para ones. And there was no halocins, but this hypothetical proteins. Probably this protein is submitted by someone in the database, is not characterized. And this halocin had a similarity with these hypothetical proteins. So we try to understand, because this hypothetical protein in the database are reported, but their functions are not known. And we thought that we should understand what these sequences are. And we analyzed these sequences and found that the higher number of negatively charged protein amino acids are present. And because of the presence of high number of negatively charged residues, the whole net of this net charge on the protein is negative. And this is responsible for the halophilic nature of the molecule. So it creates a non-bulky group and also a high number of negatively charged amino acids. That actually gives a kind of conditions where it is able to tolerate, able to resist high concentration of NaCl. So in nutshell, I would like to conclude that these haloarchaea, which were able to isolate it from this salt lake, we say it's a Pachpadra salt lake, and they are the molecules which are being secreted by these haloarchaea or halocins, antimicrobial peptides. These haloarchaea are able to tolerate high salt concentration and so strong pigmentation carotenoids. So it's one of the halocins we have tested. They are having a broad range of stability, not only high temperature, pH and NaCl, but also in the organic solvents. One can understand that if you are having that kind of stability, then one can select the range of application, either in the food or medicine. We need in both the sector that such kind of extreme conditions, if any molecule is stable, one can use. The activity of both the halocins are completely salt dependent. If you are having a particular more than 10% salt is required for the antimicrobial activity, and molecular weight is approximately 14 kilodalton. Multi-top MS analysis and N-terminal sequencing both indicates that the molecules are unique, and its structural analysis shows that it does not belong to any halocins which are present in the database. So this study gives an opportunity to the researcher that we are not only having a halocins, but the cell having several metabolites, either it is intracellular or extracellular. We have experience with the extracellular in this case, but even if you see the extracellular secretions, there are not only halocin, but so many, more than 50 peptides we can see on that SDS page. What are others? We need to concentrate on one by one to understand why and how these archaeal strains are secreting such type of molecules in the nature. They are exotic or spatial, or we understand both, we don't know. So there is a completely kind of opportunity where people can work. It's open to all. I have submitted this culture to the general database culture collection center in India, which is available to throughout the world. Anyone can take it and work on the different aspects to solve the problem what we are having related with the food and medicine. As I told in the beginning that my laboratory is mostly involved on the probiotic bacteriocins, but in one of the aspects we started working on the halocins. Out of these, we are having 5 molecules in our laboratory characterized in last 30 years. We are involved in this. One is plantaricins and enterocins. We have isolated these two from food samples. In India, we have very indigenous fermented foods. So we isolated from these two, and pediocin and visilacins we have we are having these from soil isolates, and have a very good agricultural application. One can go for that. And plantaricin and anthracene can go for food and medicine application, whereas halocin, H1 and H3 we have already discussed with you. And right from the beginning, from the structural point of view, stability, antimicrobials, mode of action, these parameters we are characterizing in the laboratory, how to have this unique molecule, how to having the features which are actually required by the industry and which are lacking from their things, and it can be applied for the food safety as antimicrobials. We are having both the situations are problematic throughout the world. In the food, our regulators are very strict, but even after that, we have found that in some of the country, there is a huge disturbances of the addition of toxic substances. What we say in the junk food or something, they're adding the preservative, chemical preservative, which are very toxic in nature. Sometimes it's a carcinogenic and sometimes it's a very allergic. And people are eating since throughout the world this junk food, or you can say the chemical preservative we are adding in the food. So it's one of the important aspects where in the food there are so many toxins are being added, and they are causing several issues, several diseases are synergized. We are having a new type of diseases which were not available earlier, because since last 30 to 40 years we started eating packed food with lots of preservative in the high concentration, and the new research has reported that it has several side effects, and people are eating. I have seen most of the developing country, there is a lot of problem of regulation, and these regulations are not that strict. Same way with the antimicrobials. My friends, trust me that the world is suffering with the antibiotic resistance. A day will come, it will be more like a COVID, more like a corona situation we are heading. And, patients are dying in the hospitals, but clinicians have no options to treat, because most of the antibiotics, what they are having is bugs have already developed resistance against this. And this is just like a presentation, but you think of the message, that what kind of message we can give to the society, what we can contribute to in these two areas, whether it is a antibiotic issue, resistance issue, or addition in the food, adulterants or food preservative, whatever chemical based preservative they are adding. So we have taken these two aspects as a target mission to solve the problem of the humans to the society. We are moving ahead to the microbiota modulations also, because it's a new feature of bacteriocins and probiotic. They're able to modulate gut microbiota and others. And by modulating the microbiota, you can change the disease condition from healthy to disease, from disease to healthy. And I mean, if a person is diseased, it's a gut microbiota is different than the normal. And if you correct the microbiota, the individuals become healthy without any chemical treatment or without any medicament. We have seen this. Yes, I forgot to say that the halocins we have seen, it's a potential antioxidant. And in one of the study, we have purified these carotenoids from the haloarchaea also. They are showing an extremely very good antioxidant potential. And the efficacy of antioxidant activity is much more higher than the commercially available antioxidant like ascorbic acids and others. So these features we have characterized in our laboratory and found to work on this by the supports of several funding agencies from the national level. We got it, and we have addressed this issue to several platforms, to several journals, and they encourage that yes, this kind of work should go ahead and may solve the current problem we are having throughout the world. This I really like. I have not done these works. Left side, my students, they were involved and help from all the collaborators from this Professor Michael Chikindas and Professor Todorov is also working with us, and we understand, we share a common interest and found. So some of them, they are involved in the isolation, characterization, purification, antimicrobial, antivirals, antioxidant, oral health. So these we lost this friend, Professor Deep from Norway. We met in the Prague in the last conference, I think. So thank you very much for your attention. I hope I convince you. If any question, I'll be happy to answer.
Thank you very much for the interesting presentation and giving the lights on the different group of the microorganism producing bacteriocins. And in fact, we have already some questions on the chat system. I will start with the question of Gabriela that actually you partially answered in your presentation. If bacteriocins from archaea microbes can be active against some gram-negative, but you already mentioned in your presentation that some of them, most of actually, are active against other archaea, other halophilics. So is any exception, some of the this bacteriocin that you studied, they had activity against gram-positive or gram-negative bacteria? Or was again the same dogma that closely related are much more affected by the effect of the bacteriocins?
Yeah, and this gives an opportunity that if you are having a narrow range spectrum, and that will be specifically utilized to remove such type of haloarchaea contamination from the food. Most of the salted foods, they are having haloarchaea contaminations, and the hide leather industry, there is an involvement of haloarchaea. I mean, this kind of study from the it's a kind of intercepting study from bacteriocins to halocins, and we are not having tested even the cocktail, the synergistic effect of halocins and bacteriocin. And one can select the range of bacteriocin, which one is effective against, what is your target? I mean, you have to decide what is my targets, and the targets are completely the application based. So if we have to go for this target, one can select the bacteriocins and the halocins and can assay it separately or in a combined mode to reach to the target level, whether it is being killed or not.
Thank you. Then Iker Oliveira was asking actually several questions. First of them, is the halocin production a phenomenon associated with quorum sensing?
Oh yes, not worked a lot, but in case of archaea, it's not reported, but in bacteria, we know it already. Yes, they do follow this pattern of quorum sensing pattern, where we in our laboratory have not tested, honestly, but I think there should be.
So it's still a lot of things to be done in this direction. Then Iker as well asked, what are the potential resistance pathways that target microorganisms may develop against halocins, and how the haloarchaea responds evolutionary to these adaptations?
Yes, it is a big
problem. So how this can be?
Yes, it's a very good question. If you compare with the bacteriocins, like antibiotic, they are developing resistance easily. But in case of bacteriocins, bugs are not able to develop resistance in that extent, because we already understood the target. It's not only a membrane, but most of the time these peptides goes inside and target the cytosolic targets are there, the proteins and the enzymes inside the cell, and sometimes they disturb the DNA and RNA replications. So it's a kind of multimeric targets. So like antibiotic, they are having a very specific target, and bugs are developing resistance against that particular target by modifying it. But in case of halocins and in case bacteriocins also, developing resistance is very low, or sometimes it is not possible, because in one of the case, the cells is physically disrupted. If you kill the cells physically, cell is not able to, because it's a membrane disruption, mostly in case of halocins also. By disturbing the sodium hydrogen antiporter, it's like a membrane disruptor, and the cell is not able to develop a resistance against the halocins. With the very less report, we are working on it, but we have not found any resistance development against the halocins. So it's a positive sign that we can replace our antibiotic with these combination of halocins, bacteriocins, by optimizing the conditions, by optimizing the molecules from the different sources and different types. Maybe it can give a synergy with the bacteriocins. If it is not inhibiting one target, you can use bacteriocin, halocins together, may work fantastically.
And do you think the halocins will have same way of mode of action or different mode of action compared with this one from gram-positive and gram-negative bacteria?
Oh yes, because the content of this peptide is entirely different than bacteriocins. Bacteriocins are positively charged, mostly cationic in nature, whereas this one is anionic. And in one of the case, like halocin H6, is reported that it targets sodium hydrogen antiporter. But this kind of a study is not reported in bacteriocin. So the mode of action is entirely different for halocin. This is what we think of, that if you are having two molecules, its mode of actions are different, then it will kill the target very easily. So this is how synergy is achieved, as we have seen previous study.
Okay, any more questions for the audience here? Okay, thank you very much for your presentation. It was really a pleasure to be with us today. Sorry for late time in India that you needed to present, but we have quite a big time difference. Here it's 2 o'clock p.m. What time is there?
It's 10:30 in the night.
It's quite a big difference, but thank you very much to be with us today. And please give him applause, because it was nice.
Thank you, Dr. Todorov, for inviting me in your conference. Thank you very much.
Thank you very much, and see you in the next occasion. I hope very soon. Thank you.
See you. Bye. See you. Bye.
Bye-bye. Well, next presenter have a technical problem. He's lost his voice. And he apologized. He cannot be presenting today with us, but we're having a final kind of, not closing remarks, but a very important presentation from Christian Hoffman. He's at the moment, how I can say correctly, it's taking care for the international relation in our faculty. There must be specific word for this. Chair of the International Relation Committee. Sometimes I say director for the international relation, but well, depends of the interpretation. And this event was sponsored and organized by Alkani. And this is actually touching point between international relations of the university and International Relation Committee of the faculty. And it's interesting to have Professor Christian Hoffman today with us, because he will give some information about international relations, international programs, how it's actually work, what we can do, how the students can go for abroad, how we can receive. So there is no need for slides for such a presentation, because it's, I mean, everything is answering question and answer. So I hope maybe there will be more questions later.
Thanks, Lavi. Thank you again for the introduction. Yeah, the word in Portuguese would translate as president, actually, but normally we would use chair, I guess, or director. Yeah, chair or director. So yes, as Lavi said, I'm chair for international relations at the School of Pharmaceutical Sciences here at the University of São Paulo. We do all the field work, so to speak, and actually maintaining and organizing and giving support for all sorts of international relations at all levels, from simple internships for undergraduates all the way to dual degrees and dual diplomas for courses in the school. So I think that the first thing that I would like to say is that people should keep in mind that things are usually simpler than they think. They are not so complicated. University of São Paulo has a decentralized system. So although this event was sponsored initially by the Central International Relations Office, ALCANI is the acronym that we use, each one of the schools within the university is independent to strike agreements in any way the school sees fit. The University of São Paulo has a federated system. So all the schools have independence to be able to decide how they would like to pursue their goals and missions. So at the School of Pharmaceutical Sciences, we're free to actually strike agreements with other schools, should they be able to do that with us, or with other universities, if that is the case. And normally, it is preferred to follow that format because the interests tend to be a bit more narrow, or it's a little bit easier to actually keep the relations more egalitarian, right? For undergraduate students, this tends to be formal exchange programs that we have with other universities. For graduate level programs, this is a very, very simple process. It really depends on the students and the groups, the research groups, getting in touch with each other and actually starting the conversation. And we're here to help, and we're here to actually give the support that's needed to make that happen. At the moment, we have at the University of São Paulo, at any given moment, we have a very, very large number of students visiting us and actually following degrees full-time or just as exchange programs. And at any given time, we have just about the same amount of students actually leaving the University of São Paulo now, fortunately, after the pandemic, coming back to the normal numbers. At the university, at the School of Pharmaceutical Sciences, we have 4 degrees that people can actually get in touch with, 4 graduate programs that actually can get in touch with, depending on your research line, depending on your approach to research. The one that's most pertinent here probably is the newly rebranded and renamed program of integrated food systems that actually is a very large effort for the University of São Paulo to actually bring together all the expertises around food systems, all the way from production in the fields, all the way to post-consumption and health effects and everything in between. And that reorganization is really redefining how we think about that and being pioneering within Brazil to address the real need to understand the entire cycle of production in consumption and health effects of what we actually ingest. So it's very appropriate, I think, to mention that a very large component of this program will be offered in a hybrid mode. So we'll have classes that would be, physical, but we'll also have it broadcasted. So this is actually an effort to try to, bring more people to University of São Paulo to be able to open the possibility for people to actually interact with our staff and with our faculty. So if you have any questions that are specific regarding to exchange programs or visiting, do not hesitate to get in touch with me. My email was listed earlier in the week on my presentation when I was talking about my research specifically, some research specifically. But of course, you guys can also get in touch with Professor Todorov to be able to, be forward to the international relations, and we can actually certainly put you in contact with the right people to actually make sure that you visit or you exchange or eventually even come full-time to the University of São Paulo. We are open to all levels of interaction. So I think that we have a little bit of time, right, Slav? So we could see if anybody has any questions specific that I can answer right now on the chat or here in the room. I'll be happy to oblige. We do, but that's at the undergraduate level. Yes, we do have a formal agreement with, for exchange between undergraduate students with France, University of Marseille. At the moment is the one that we have focused more heavily, but we're expanding this to actually include other universities within the next years. We have the intentions of actually at least 2 more agreements of this kind. And that is a formal agreement. So students are selected here, they're selected there, they'll come back, they will stay here for a semester, they'll take classes. That is a very much undergraduate heavy kind of effort. At the graduate level, this is much more simplified procedure. It really is contact the groups and then decide if you would like to take a class or if you just want to do research. It is a much more flexible dynamics to actually be able to work with. For undergraduates, this tends to be a little bit more complicated, right? Because you have classes and you have the semesters that you have to keep track of. So that requires a bit more planning. But it's possible. And it's possible outside of the formal agreements as well. We're willing to accommodate whenever possible. All right. Well, if there's no questions at the chat or from you guys, thanks so much for this attention. This is really, it's been kind of an interesting week actually watching it all behind the scenes and seeing everybody presenting. So yeah, thanks.
Thank you very much for the information. It was really nice. Well, with this, we finish. This was the last presentation of Christian. So, but don't think that you escape forever. We will call you in the next time when you have the seminars to give the more clarification about everything, not only about the programs that the university is involved, but as well a few personal research because, well, bioinformatics is a very interesting tool. Even if I don't believe fully in bioinformatics, we need to use it because it's giving a good answer and facilitate the rest of the work. So everything combined is making stronger of everything. So thank you very much for all the participants that been here in the Faculty of Pharmaceutical Sciences, everyone that survived watching from all around the world. And in next week, we're going to have starting to preparing the certificates for the people that presented. We've been in presence in here and in online. We have all the information from the your participation chat when we confirm your participation. So thank you very much again for Aukani for giving this option to organizing and to sponsor this initiative and coming with this idea earlier. Actually, end of last year, they started everything and it's taking time because everything needs to be organized. Confirmation of all the presenters from all around the world. And thank you very much for all the presenters, and hope next year we're going to have the similar course with the different subjects and different presenters, and to try to enrich our knowledge of everyone. Personally, I learned several things, and I hope you as well. So thank you very much, and see you again next year. Thank you.